Good morning. I think everyone is still feeling the heat from outside. Good morning. Okay, all right. For those of you on the webcast and for those of you up here, my name is Khoso Baloch. I'm the CEO of CorMedix. Delighted to have all of you here, either physically or on the webcast, and you taking time to be with us so you can see some of the developments CorMedix has done over the last few months. Before we dive into the presentations, what I'd like to do is outline for you the strategy of where CorMedix is going. So if you look at the strategy, it is under three main themes. There is the foundation segment, which is on your left side, where here we look at ensuring that we have favorable financing and couple this with the greatest inflection point for the company, which is Neutrolin, completion of the registration studies, getting the approval, and then launching it successfully in the United States. And that's what we call the foundation and the biggest focus of the organization. The second column is partnerships. And for a company of our size, it's critically important that we leverage partners to give us the bandwidth that we require. And here we look at partnerships in terms of our supply chain to ensure we've got an agile supply chain, favorable, competitive cost of goods, but also looking at where we have approval outside of the United States for Neutrolin and using partners who understand the hemodialysis segments and the hospital segments to ensure that we can leverage Neutrolin in those markets. Then the third column we call leveraging, and this is taking taurolidine platform and ensuring that we can find other solutions using the medical device pathway, which is the 510(k) pathway, or the NDA pathway, because of the properties that Neutrolin has. Our focus today will be very much on the third section, which is leveraging. We have spent a lot of time talking about bucket number one, but it's bucket number three where we've made tremendous strides over the last few months. So a big part of the focus will be there, taking the incredible properties that taurolidine has and looking at the 510(k) pathway and ensuring that we are able to move very quickly, very efficiently, and be able to deliver new solutions in some very large segments which are very, very interesting. We will spend some time also on the foundation because there are some studies that we have done on the side to our main phase 3 studies that we would like to share with you some very interesting information. So that will be the focus of our discussion today during this meeting. For those of you on the webcast, you will be able to submit any questions you have via email that has been provided to you, so we will be able to answer it. There will be Q&A at the end of each section and also at the end once all the sections are completed. Now, for the moderator of this meeting today, I would like to introduce Gary Gilfish. He needs no introduction. Many of you already know him. He's been practicing vascular surgery since 1990. And as we've been having more discussions, finding out he's been also giving lectures to some of you who have been here. His exclusive focus is in the hemodialysis section. So the care, some of the challenges, complications for hemodialysis, he's got first-hand experience. Widely lectured and also published. So with no further ado, Gary, if I can introduce the floor to you. Thank you.
Thank you, Khoso. I'm really glad to be here. Okay, it's exciting for me to get away from the drudgery of operating on patients and look to the future. And the future is indeed bright, and we'll talk about this. Am I pointing? Let's see. There we go. Okay, this is the structure of taurolidine. Looks fancy. Okay. The breakdown product of this is actually taurine and some associated compounds, but this is the compound that we'll be talking about. And I'd like to get into a little bit of history. This is actually the first commercial advertisement for penicillin that appeared in 1944. My father, actually having gone through World War II, was in a hospital for a couple of years because they didn't have antibiotics, and the only way to take care of wounds was basically to let them fester and open, and the pus would come out, all these wonderful things. But the interesting thing about this is this was in an era of great excitement for the new wonder drug. And penicillin, invented or discovered in 1928, took almost 20 years to commercialize. The federal government contracted with 20 companies to try to get the supply up. So only by the end of World War II did enough quantities come out, and essentially it was reserved for soldiers. I was born in 1957, and that was the peak of the penicillin craze. And when my sister had a sore throat, I would get a shot of penicillin in my butt, okay, because it was just a good thing to do, so to speak. So what's wrong with that? What's wrong with the wonder drug of penicillin? And it became very clear, even in Fleming's time, who discovered penicillin, that this is a drug, and bacteria are pretty smart, and they figure out how to get around it. This is an interesting slide. Do we have a laser pointer, by the way? Oh, I see. Okay. Where no sooner do they come out with a class of antibiotics, okay, for example, penicillin, by 1940 they had developed strains that were resistant to it. And these are the different classes of antibiotics. They come out with one in 1948, and by 1953 we have resistance to it. It is so bad at this point that many bacteria have multiple resistances to different classes of drugs, and in addition, for the past 30 years we haven't come out with any new classes of antibiotics, although we've come out with antibiotics within a certain class. And this is data from the FDA, which looks at the antibiotics brought to market, and you see an amazingly sharp decrease in the numbers, okay, down to only 3 during a 4-year period. And this is when the FDA and Congress appropriately were very concerned. And they said, not only do we have an increase in the number of resistant bacteria, we're also not developing new antibiotics. Okay, so this culminated in the FDA Safety and Innovation Act of 2012, where part of that was having the GAIN Act, which is an acronym for Generating Antibiotic Incentives Now, and there was something called the QIDP, Qualified Infectious Disease Product, which Nutrilin was granted, and it gives certain benefits. So I just want to give the overview of where CorMedix is. If you're a bacterium, as Mark Twain said, you know, reports of my death are greatly exaggerated. You know, bacteria had a hard time in the mid-40s, but they've since said, hey, not so fast, guys. We're going to try to outsmart you. If you're a patient, a public health officer, an infectious disease microbiologist, you're getting a run for your money now, okay? Because this really is a problem. But if you're CorMedix or another company that's dealing with emerging technologies and exciting emerging technologies in this market, there's tremendous opportunity. You know, the medical field is starving for the type of innovation that we're giving them. So I just want to review today's agenda. We'll have some basic science overview of taurolidine. We'll talk about taurolidine incorporated medical device. This is as compared to the LOX solution, which is really classified as a drug. We'll talk about some really interesting stuff in terms of taurolidine-based therapy for cancer. And last but not least, and we're putting this at the end because you've heard it before, and we're most advanced in this area, is Nutrilin, our flagship product, which is for prevention for bloodstream infections. We have a highly qualified group of people who are going to present, and I'd just like to go through them for starters. Tony Faffel has been with us for about 10 years. He was a co-founder of the company, board member, and now he's Chief Scientific Officer. And Tony has held many other positions in biotech companies, including Director of Healthcare Research at Baring Capital, Managing Director at Paramount Biocapital, and Principal and Founder of Black Diamond Research. Before fully engaging in the biotech industry, he was a nephrologist practicing in New York Hospital, Lenox Hill, and Memorial Sloan Kettering. Tony has pushed the dream of bringing Nutrilin to the market for longer than anybody else. I think it's appropriate to say that, and he'll be presenting some key lectures today. Our second presenter in the first part is Robert Bob DeLuccio, who I've had the pleasure to meet over the phone several times and now in person. He's an experienced chemical and biochemical engineer and has extensive experience. I'm cutting down these bios that you have in front of you just to get the salient points. He spent most of his career at the DuPont Experimental Station, where he developed polymeric drug delivery systems. And now I know those patches that I prescribe, where they come from. Thank you very much, Bob. Okay, including the first transdermal drug delivery system capable of delivering pain medication. Prior to that, at Ethicon, Johnson & Johnson, he led the suture group responsible for research and development of resorbable and antimicrobial sutures. So Bob also has 35 issued U.S. patents. And currently, we are fortunate that he serves as our Director of Research and Development. So Bob brings a tremendous amount of research and practical experience that we seldom see in one lifespan. So, and he'll give us some insight there. Is Dr. Lawrence here? Yes. Hi, Dr. Lawrence. We haven't met. Dr. Lawrence is a world-renowned, board-certified aesthetic plastic surgeon, and is certified by the American Board of Plastic Surgery, a fellow of the American College of Surgeons, and a member of many other professional societies. Besides establishing his private practice in 1992, Dr. Lawrence has been internationally recognized for his research and development and for his advancement of new aesthetic surgical and non-surgical techniques. And perhaps you'll give me a curbside consult about my bags under my eyes when I'm done here. Just kidding. I was hoping to avoid that, but it's okay. So Dr. Lawrence regularly contributes to many lay and professional journals and publications. He is an author, patent holder, and innovator, and we are really glad to have him as part of our Suture Development Group. So without further delay, I'd like to introduce Tony. Go ahead.
Thank you very much, Gary, for that introduction, and it's a pleasure to have the opportunity to address the audience today, investors, physicians, researchers, and people who have been very helpful to the effort of driving this process of bringing Nutrilin and Taurolidine-related products to market. I'm sorry. Okay, so Taurolidine, we want to review the mechanism of action of the drug, which is important. How does it work? It works in many different capacities. If you look here, it's not an antibiotic, it's an anti-infective or antimicrobial agent, and it's been shown to reduce the infection rate by up to 96% compared to benchmarks in our post-approval surveillance study in Europe, which essentially has been published in several peer-reviewed publications, and we have a third publication which is in process. It's also an anti-inflammatory, and it helps prevent inflammation, and this was the initial application as a lavage solution in surgery in Europe, and it reduced adhesion formation, which is a complication of surgery. It prevents biofilm formation by disrupting cell walls, and also the bacteria which are enmeshed in the biofilm. This is key, and you'll be hearing a lot more details from Dr. Greg Schultz, who is a biofilm maven and runs the Biofilm Center at the University of Florida, Gainesville, and also acts as an endotoxin. It binds and neutralizes bacterial endotoxins, lipopolysaccharides, etc. The antimicrobial, to scope in on the antimicrobial properties of taurolidine, it's active against, the beauty of taurolidine is active against a wide variety of organisms. Gram-positive organisms like Staphylococcus and Streptococcus, gram-negative organisms like Pseudomonas, and it also works against fungi, and you'll hear a little bit more about its effect against not only Candida albicans, but Candida auris, a particularly virulent bad actor in the infection environment in hospitals and in patients with catheters. It also works against resistant bacteria such as MRSA, VRSA, VRE. How does it work? It essentially works at the level of the cell wall by blocking the cross-linking of peptides or proteins that make up the cell wall. It prevents biofilm formation in addition, and it also disrupts the attachment of the bacteria. Some bacteria like E. coli have processes called fimbriae which attach to the endothelial cells, and the taurolidine actually competitively competes with mannose to block attachment of the bacteria to the human endothelial cells. So it works through a variety of mechanisms which we feel all add up to reducing the ability of bacteria and fungi to form resistance against taurolidine, which is the active component of Neutralin. We are focused currently on multiple facets of its mechanism so that we can leverage them, as Koso said, to develop not only Neutralin, our flagship product, but also products that contain taurolidine to help fight infection, inflammation, and tumors, cancer. Okay. Here's a slide which many have seen before that shows the role of taurolidine in preventing biofilm. Here's a photograph of biofilm that develops if untreated within 24 hours. If you use heparin alone without taurolidine, after a period of 7 months, you have a lawn of biofilm, very dense biofilm. If you use Neutralin with taurolidine, you see that largely the biofilm is inhibited from forming here. It's a significant unmet need. There are about 250,000 catheter-related bloodstream infections per year in the U.S. Biofilms are responsible for a majority of catheter-related bloodstream infections. And if a bacteria is in this lawn of biofilm, it's enmeshed in this matrix over here, then you have a much harder job of eradicating the bacteria and fungi because it's in that lattice work. So it's very hard to get rid of it. So by inhibiting biofilm, that helps us get rid of the bacteria and fungi. And you can see this is not a trivial problem. The mortality rate is about 12 to 25%. And these are very sick patients with multiple comorbidities, other diseases. Okay. Anti-tumor properties reported in the literature, and this is the reason why we're pursuing the oncology indication. It's broadly active against diverse tumor types. So it's not only our first target, which Dr. O'Neill will discuss, neuroblastoma, but also other rare pediatric tumors and adult tumors such as lung and breast cancer. We'll get into more detail of that later. It enhances oxidative stress, and this is shown by, if you use glutathione, glutathione inhibits the oxidative stress pathway, and it can be attenuated by glutathione, which proves that glutathione S-transferase, which is the enzyme involved in that pathway, highlights the fact that oxidative stress is enhanced by using taurolidine. It induces programmed cell death or apoptosis. That's the terminal attack sequence that kills cancer cells. It also inhibits vascular endothelial growth factor, or VEGF, induced angiogenesis. It's reported to be highly selective for tumor cells as opposed to human cells, which reduces the toxicity, and our delivery mechanism lends itself to that. It also reduces inflammation as mediated by commonly known cytokines, interleukin-1, interleukin-6, and tumor necrosis factor. and it has an ability to show drug synergy in combinations. As you'll see later when we report some of the early studies which we have conducted, it works synergistically with other anti-neoplastic agents such as the vinca alkaloids, vincristine. Okay. Neutralin, if we look at its clinical validation and safety, we have conducted a Neutralin use monitoring program in Europe that has been conducted at 22 sites in Germany, and it involved 202 patients. In those patients, there were no serious adverse events reported, and if you look here, you can see that there was a reduction compared to historical controls by infection by 96% and thrombosis by 96.7%. We're looking to validate this in our prospective study going forward, Lockett 100, but this data, albeit historically controlled, is very encouraging. It was, again, 202 patients, and the number of catheter days, the number of days patients had catheters was 36,000 roughly. Again, in the EU, no major adverse events were noted. Okay. Now I'm going to turn over the program back to Dr. Gelbfish, and we will proceed.
Thanks, Tony. So the next topic is antimicrobial sutures and meshes incorporating taurolidine. This makes a lot of sense. If you have an antimicrobial, use it wherever you can. And the reason for that is antimicrobials are different than antibiotics, where antibiotics typically are given systemically. You give it either intravenously or orally, and it makes its way around the the body, of course. Antimicrobials are put on the site of infection to prevent infection. And if taurolidine has been shown, not if, but considering that taurolidine has been shown to inhibit growth of so many bacteria, why not incorporate it into devices or things that you implant so that they won't get infected in the first place? Because treating is what you want to avoid. You want to avoid treating infections and instead prevent them. So let's talk about surgical site infections. There are about 50 million surgical cases a year in America, and a rough number is 2% of them become infected at a tremendous cost. Now, a lot of them will get infected regardless. I mean, if you have significant trauma, or if you have a dog bite or something like that, I mean, you're not going to change the infection rate based on the suture you use. But there are many of these infections where using a suture, which is a foreign body, that by itself can enhance infection rates. So what you want to do is potentially change that by incorporating taurolidine or another antimicrobial into the suture itself. Now, this concept is not new, and that's the critical part here. There is one other option which only came on the market in 2003. It's a product by J&J. It's called triclosan. And even though it's only been on the market for 12 years or 13 years, it already has made such significant inroads, and I'll show you the actual language, that both the World Health Organization and the CDC currently recommend its use under certain circumstances. So the concept of incorporating an antimicrobial in all types of sutures and meshes is not something that we're proposing the first time. It's something that is well accepted, again, despite the fact that the concept was only developed a little more than 10 years ago. Now, the other option, okay, this is not talking about competition. It's always good to have two options. And besides, triclosan is something that's undergoing significant regulatory review by the FDA, and it's something that is well recognized that an additional or substitute antimicrobial would be useful. This is actually the World Health Organization in 2016. They go as far to say and this is verbatim, the panel suggests the use of triclosan-coated sutures for the purpose of reducing the risk of surgical site infections independent of the type of surgery. They're basically saying all sutures should be antimicrobial, okay? I can tell you today, I don't have exact numbers, I've never used an antimicrobial suture. That's how little the inroads are, okay, over the past 10 years. But somebody in the World Health Organization, this committee, is so concerned about infection worldwide and so concerned about SSI that they're making this huge statement. This is something that just came out in March of this year. Centers for Disease Control and Preventive Guidelines for the Prevention of Surgical Site Infections. They're saying consider the use of triclosan coating sutures for the prevention of SSI, meaning use your judgment, see if the case is one that has a higher likelihood of infection, and use this. So this is something that has fallen from heaven, so to speak, you know, into our perspectives. Obviously, the company has been thinking about this for a while, but as you'll see from the next presentation, the company has a very active program to try to develop this, and Bob will be sharing with us his findings. Thank you.
Thank you, Gary. Thank you for your kind introduction before. Just want to give you a little flavor of what we've been trying to do. Previous to joining CorMedix, I was doing research and development mostly in drug delivery systems, medical devices, and other applications. I did the drug delivery work mostly in DuPont, and then a lot of the medical device work at Johnson & Johnson. When I was at Ethicon, we didn't only work on sutures, we actually helped the other J&J companies make other devices. I even worked on the Cypher stent, so I had a lot of interactions with Alza Corporation, if you ever heard of that company. And when I was at DuPont, I actually developed a transdermal delivery for the first opioid that was ever delivered through skin. And I did work on the fentanyl patch. So J&J still sells that patch. So I did a lot of things, a lot of interesting things in drug delivery. So when I was asked to come to CorMedix and look at taurolidine, they were saying, don't you think we could put this antimicrobial in some devices and do something with it? Well, I thought about that. I said, why not? It's a non-antibiotic, anti-infective, it's anti-inflammatory, prevents biofilm formation, and it's an anti-endotoxin. I never found an agent like that before. Why wouldn't I want to put this in a medical device? So, if you look at it as a franchise, you do have drug opportunities, and you also have medical device opportunities. We're looking at going after neuroblastoma, osteosarcoma for the drug opportunities, and in medical devices, sutures, topical hydrogels, and meshes. Now, to talk about sutures a bit, there's a collaborator I had when I was at Ethicon, not for sutures, but for soft tissue augmentation. And that happened to be Dr. Lawrence, who's sitting here today. He was hired by us in J&J to be an advisor in this area. So he and I have had a long relationship, I think 10 years now, Paul. So when I first saw taurolidine, I asked Paul, do you think this thing would be good to put in a suture? And he said, yes, I'm a believer that it's a good idea to have whatever protection you can in the product you use for surgeries. This is especially true for plastic surgery. He also brought up the fact that with bariatric surgery becoming very popular, there's lots of sewing that's done, so it makes sense to have something there as insurance to protect against infection. So he and I decided, let's write a patent and cover this thing. And we never did any experiments when we wrote the patent. So I said, we'll try to get the data and see what happens. I don't even know if it's going to work. So we wrote the patent before we knew anything about it working. But it meets an unmet medical need. Antimicrobial sutures inhibit surgical site infections. Current antimicrobial sutures use components with known adverse effects and environmental impact. In fact, a very good friend of mine runs the group that's looking for the alternative of triclosan and Ethicon today. I never mentioned this product to him. So and I don't want to until we know we can get something that's useful. So there is a government movement to remove products containing the components, and FDA did make an edict that some over-the-counter products can no longer contain triclosan. So, talk about how much of a market this is. There's 40 million cases. There's about 300 to 500,000 surgical site infections predicted to occur annually. We really don't know this number accurately. And if you were at a J&J update in terms of earnings, they never reveal how much of their particular products are sold. In about 19 updates that I witnessed in the company, I never knew how much of a market the antimicrobial sutures had because they wouldn't tell us. So, in terms of feasibility, I created prototypes to demonstrate the effective antimicrobial activity. We looked at potential predicates, because if we want to approve this through the 510K route, we do have to have predicates that exist. Since J&J did approve their product via a 510K, I can use their existing products as predicates. So as a result, we would have an easier time filing for approval with FDA once we get appropriate data. So the planned milestones are as follows. We're doing feasibility with refined prototypes, which I did recently. We actually created a PDS plus type suture using commercial type manufacturing equipment to make an identical product to PDS plus. So that I recently did and actually had it tested recently. There's Professor Schultz in the audience here. He is the one who's done all of the evaluations of every device concept I created. So I give him a lot of credit for making me honest, because he would tell me if it wouldn't work. So next thing we're going to do is a proof of concept animal model. And I have to thank Dr. Lawrence, because he's leading this effort with Hackensack Medical Center. So we plan on doing an animal trial at that center, and he's directing what we're going to do in terms of a protocol and the procedure we want to use to make a surgical site infection and test our suture against it. Okay. So we do have a patent that's filed. There's a PCT that was filed August 18th, 2016. And because it was filed then, this thing's already published, so the public knows about this patent. It is a patent application. It is not an issued patent. It still has to be prosecuted through the patent office. So we haven't gotten any adverse office actions on it yet. It's still going through the process. Okay, the next thing I want to talk about is non-woven meshes for hernia repair. But realistically, these things can go well beyond meshes for hernia repair. I'm sure you guys heard of pelvic floor repair devices made with meshes and other applications. So we want to bring up hernia because it is an attractive market to hit. The unmet medical need right now is there is no marketed resorbable antimicrobial synthetic mesh product currently on the market. There are semi-resorbable meshes out there. Usually they have a base of polypropylene that lasts until you end up in the ground. So it has degradable and non-degradable components in it in the most advanced type mesh that is out there today. I'm more interested in meshes that will go away eventually and let the tissue heal itself. So for this reason, I will use resorbable products that last a long time to allow tissue to repair itself. So the current available options, like I said, consist of permanent or living tissue. There are additional needs in the prevention of infection and hernia repair, as well as other soft tissue applications. I don't know if you know this, but Ethicon is also in the mesh product area where you introduce meshes to help hernias. The product we launched while I was there was called Proceed. It was semi-resorbable, and the end product was to end up with polypropylene with the cells integrating themselves around the polypropylene mesh that was left over. The unfortunate thing is, if there was an infection or failure, those products would extrude out of the body of the patients that have it. So this was a complication we never liked. So that's why I always thought, why not make a fully resorbable mesh? You'd be better off. So there are mesh prototypes to demonstrate highly effective antimicrobial activity. There's about 1.5 million products out there. That many surgeries are done a year for hernia repair. It's estimated that about 400,000 of them fail one way or another, and this is not an unrealistic number. It turns out that unless a procedure is done perfectly, you can expect to have a problem during a hernia surgery sometime in your life. So, and it's not always due to how it was installed. A lot of it has to do with how well you treat yourself once you have this mesh implanted in your body. If you're very aggressive about exercise, you could actually dislodge it while it's curing. So, we have created these things using electrospinning. One of the nice things about electrospinning is when you do create the fibers that way, you actually take a solvent, dissolve your polymer, you dissolve your active ingredient or suspend it in the solution. With a high voltage electric charge, fine fibers come out. These things create non-woven meshes. I did a lot of electrospinning when I was at the DuPont experimental station, so I did find researchers who have capability on making good electrospin meshes. I use Professor Gary Bolin, who's at the University of Memphis, and he's the one who created these mesh concepts for us. So any of the data you see related to meshes are due to him. Any of the evaluations of the meshes in terms of how they perform come from Professor Schultz. Okay, so we did have a patent filed as well to cover these concepts. That was filed in August of 2016. That patent also published. Okay, so now we looked at another area, hydrogels for burns, decubitus diabetic foot ulcers and wounds. Actually, hydrogels came up early before I came to work for CorMedix. I was asked, why don't you look at hydrogels and see if you could do anything about wound healing? The first one I thought about was one that's popular out there. Dr. Lawrence and I worked a lot with hyaluronic acid, and in fact, he and I worked on a new concept to have a long-lasting filler developed with hyaluronic acid. So I said, why don't I try to put this active ingredient in hyaluronic acid? Well, I did that, and right now, there are no current antimicrobial agents that have an agent like ours. They usually contain silver, chlorhexidine digluconate, and other materials. You'll hear more about some of the current materials used today, but when Professor Schultz gets up to talk about biofilms, but still, silver always became an attractive addition. There are some lousy things that happen with silver. It often turns black in the area where wounds occur. Once it interacts with blood, it looks terrible, and people hate the way it looks. So, although it's effective, it's not very attractive. So, we developed prototypes, demonstrated effectiveness even against mature biofilms, and that's also thanks to Professor Schultz. He has a model of mature biofilms that he's going to discuss later. That's a difficult model to come up with a material that's effective against it. So there are a number of potential predicate devices out there as well, so it allows us to continue going with a 510K path for this, and it has a potential high profit margin. And once again, we're going to be doing a proof of concept animal model that a colleague in Professor Schultz's lab is going to do for us for burns initially. So there are 187 million incidences worldwide for wounds, foot ulcers, and this is a major problem because the lifetime incidence of foot ulcers is up to 25% of the diabetes population. And Koso was in the business of diabetes. He said that this is a real number, it's not unrealistic. So we have patent applications in this area as well. We also have compositions for the treatment of joints. I don't know if you know it, that before you have your knees repaired, there is a temporary solution to inject hyaluronic acid in your knee so you can function for a little while. But the ultimate problem is you end up having to get the surgery to fix your knee. We also, Bruce Reidenberg, who's sitting in the audience here, I have to give a lot of credit to because he and I have collaborated on a lot of patents for CorMedix, and we've come up with some interesting concepts. Well, one of them is coming up with a skin-penetrating formulation of taurolidine. So that's another interesting concept we've been looking at. Okay, so we've also done this thing for drugs for neuroblastoma. We're looking for an orphan drug indication to go after this disease. We discovered there is synergy with vincristine. What's great about this? Well, the thing is, we can deliver vincristine as well as taurolidine in a nanoparticle. There's a group that developed a terpolymer, which means it's made of 4 building block segments. When the drug is introduced into this material, it actually releases over an extended period of time. It lasts upwards of 3 to 4 weeks. The thing about this is if you put the combination of the 2 drugs again together, you increase the effectiveness of vincristine to the point where you could decrease the dose substantially. What does that do for you? Well, it decreases the toxic effect of that oncologic agent. That's a big deal. Okay, so there's about 70,000 cases of these in the US. It's not a lot, but the kids who get it, it's terrible. You'll hear about this more later today. So we have gotten encouraging IC50 data. There is potential to expand this to other oncology indications, including breast cancer and others related diseases. There is this orphan drug opportunity for neuroblastoma. Like I said before, it's intended to enhance the activity of the oncologic agent. There is an experienced external team to guide the efforts through POETIC, and some of you knew about this from earlier press releases. The planned milestones for this are feasibility with in vitro data, which was completed. The other was proof of concept in vivo animal studies, which is what we need to do next to convince FDA this is a good solution for us. We did file a patent application in this prior to having the relationship with POETIC, so we will own the right to add our drug to this oncologic agent, and we'll have a pretty broad patent protection around this. Okay, so I'm going to bring one of my great collaborators up here. He's an excellent plastic surgeon. I've been trying to make him more of an engineer, but he still leaves that up to me. So, Dr. Lawrence, here you go.
Thank you, Bob. Good morning. Thank you, Bob. Thank you, Koso, for inviting me. I'm so glad I came this morning because I already learned 2 things. One, I had bilateral inguinal hernia repair with mesh in December. So you tell me that there's a 33% chance that I'm going to have a problem. Thank you. So I'm really happy that I'm here. Number 2 is that I realized that with you having 35 patents and me having 10, I have to get going because I'm way behind you. Anyway, my name is Paul Lawrence. I'm a plastic surgeon, board certified plastic surgeon in New York. I love doing surgery. That's my passion. But my other passion is doing other stuff and doing patents and clinical trials. I work with the FDA. I consult with the FDA. I consult with companies. I'm also a little bit of an entrepreneur. My last project was a company that we took from inception, from patent to IP protection, clinical trials, FDA approval. It's the LED mask that we sold to J&J last year. It's called Illuma, or the Neutrogena Illu mask that we sold the company last year to J&J for a good amount of money. So anyway, that's sort of my side thing that I do. All right, so these are my disclosures. And again, I work with a lot of companies, and I've enjoyed working with Bob for many, many years. So why Terulidine in sutures? And the answer is two things. One is $3 billion a year. That's the first answer. And the other one is what Nixon said, which is it's all about the timing, right? So this is a $3 billion business worldwide. US is about 50% of that. As far as sutures, somebody, I think it was Bob who mentioned, what the percentage of triclosan impregnated sutures are on the market right now. The data, as you mentioned, it's impossible to gather, but I did do some snooping around. I think it's about 10% of the sales are the plus sutures, which are impregnated with triclosan. So that's a big percentage of the suture sales. Again, 3 billion worldwide, half of that is the US. And the last thing is the timing, what the President Nixon said. It's all about the timing. And it was mentioned already before, triclosan is probably going to be phased out in the US. It's already phased out in Europe. So this is a perfect opportunity to have a replacement. And what we have collaborated on is to integrate terulidine into monofilament and multifilament sutures. And I personally, I like the concept of dispersing the product through the matrix of the suture because it's eluted as the suture is being dissolved. And it makes all the sense surgically. As far as the incidence of body sort of surgery, which is skyrocketing in what I do, there's close to a 30% incidence of complications with body lifts and massive weight loss surgery. It's a very significant issue and a lot of dollars spent on treating it. So I think this has a perfect role for a suture impregnated with an antimicrobial. And again, we already talked about what's happening through triclosan. So what we have been able to do is successfully load 2, 6%, and 10% by weight of terulidine into the matrix of two different types of sutures. So one is caprolactone, which is really the equivalent of monocryl, and the other one is P-dioxanone, which is PDS. J&J sells it as PDS Plus, which is infused with triclosan. Again, this is done through a twin screw extruder, blending the terulidine powder and the polymer pellets. It's blended so it's distributed throughout the whole matrix of the product and then extruded. What we are using right now in the sort of the preclinical, and I'll get to the clinical trials, what's planned in a second, but we're using 7% terulidine integrated into, again, P-dioxanone and the caprolactone sutures. Where we told you how it's extruded, it's mixed throughout the whole matrix, and again, to me, that's appealing because it's eluted as it's being degraded. So the two two studies that we had done, is really, one is the zone of inhibition. And what it means is that 200 microliters of a early phase culture of 3 different bacteria, Pseudomonas aeruginosa, MRSA, and Staph epidermidis, is coated on 25 by 25 centimeter plates. Think of agar plates. Think back to high school. So bacteria is evenly distributed. The polymer, which is infused with terulidine, is placed in that impregnated culture, so to speak. And then what you do is you measure the zone of inhibition, which is related to the antibacterial activity of terulidine. And what was measured is that increasing the concentration from 2 to 6 to 10%, of course, increases the zone of inhibition in every single one of these 3 organisms, Pseudomonas, MRSA, and Staph epi. The other experiment that was done is looking at the absolute kill of bacteria. So the first one is the zone of inhibition, how efficient it is to kill the bacteria around it. The other one is actually counting the bacteria that's killed. And the way that's done is 1 cc of a buffer solution is implanted into a disc, and then that's impregnated with 100 microliters of the suspension of the early phase bacteria, the 3 different bacteria. And then what you do at 24 hours, you just count how many bacteria, you actually do a count of how many bacteria are left in each disc. And what was found, typically in 1 cc of that solution, there's about 10 to the 10th number of bacteria per that 1 cc. And what was found at 6% terulidine suture impregnation, meaning that 6% dispersed throughout the suture. There, in P-dioxanone, there was a 5-log reduction in Pseudomonas, 10-log reduction in MRSA and Staph epi, and as far as caprolactone, basically went down to 0, or 10-log reduction of all strain tested, of all 3 strains of bacteria. So that's powerful. We have now evidence that not only does it inhibit the growth, but it also absolutely kills the bacteria at a particular concentration. So in effect, the conclusion is that terulidine fibers resist growth of bacteria in the vicinity of the fibers. The fibers that are loaded with terulidine have the ability to kill clinically significant representative microorganisms, and we picked those 3 strains particularly for a reason, because obviously they're very common in clinical infections. Also, terulidine is an effective antimicrobial, not an antibiotic, as it was mentioned, and laboratory studies showed that microorganisms are unlikely to develop resistance because the method of action is really not, it's really destruction of the cell wall. And a little bit about biofilm in my world, which is very common, or unfortunately very common, is breast augmentation, where capsular contracture, we believe, is totally derived from biofilm, and that's something that we struggle with consistently in our practices. And lastly, that sutures, meshes, and biomaterials that incorporate terulidine may be effective against surgical site infections, which I am all for. A little bit about the next step of what we're doing is that, as Bob mentioned, Bob and I are working on an animal study at Hackensack Medical Center, where we have created an animal model for impregnated or creating an infected wound, and then we'll be treating the wound with closure with non-terulidine infused sutures, and then closing it with terulidine infused sutures, and then after that we'll probably we will follow with a clinical trial. It's a very busy ER, emergency department at Hackensack, and that's one of the reasons that we chose that particular facility, and they have a very good ability to do basically traumatic wounds that we can look at, and traumatic wounds being repaired with non-terulidine impregnated sutures and also terulidine impregnated sutures, and obviously we're going to look at the benefit of terulidine in the sutures. Thank you very much.
Thank you, Paul. Okay, now I'm going to talk a little about antimicrobial meshes and hydrogels. We know that we have bacteria killing power in vitro. I tested 3 different molecular weight versions of hyaluronic acid, and I loaded them from 1.5 to 6% by weight terolidine. I didn't do this by myself. I had Cambridge Biomaterials do this for me in Cambridge, Massachusetts. They're experts at creating hyaluronic acid hydrogels in vitro. I said to them, while you do your polymerization and cross-linking of hyaluronic acid, I want you to introduce our drug. I want to see if the cross-linking does anything to the drug at all. Well, it turns out it liked the fact that it was being cross-linked in the vicinity of hyaluronic acid. There was no problem putting the drug in the hyaluronic acid hydrogel. These graphs show you living bacteria. In this case, I'm looking at Pseudomonas aeruginosa and MRSA, 2 different colors. The dark blue Pseudomonas, light blue MRSA. Got to give credit to Dr. Schultz for this, because he did these experiments to prove whether this was working or not. 0% means hyaluronic acid itself does have some effect on killing microorganisms. Well, what does this thing do when it's got terolidine in it? Well, we couldn't even make a bar. It killed everything at all the concentrations we tested. So, unfortunately, you're not seeing bars because everything's gone. So, again, we did this in another study with mature biofilms. Dr. Schultz developed a model for mature biofilms. He's trying to get it adopted at FDA. I don't know how well that's going. But, in other words, it's a very good model to determine how materials act against mature biofilms. Well, I said to him, why don't we take one of these formulations of hyaluronic acid I created, I'm going to add a penetrant enhancer to it. Dr. Schultz developed a pigskin explant model. You're going to hear about this more later, so I'll let him describe it. But what he did was demonstrate what would happen if you put these materials in the vicinity of these mature biofilms. You could see for these 3 bars here, the hyaluronic acid itself at the 3 different molecular weights doesn't really affect the living bacteria much in the biofilm. But at the 3% concentration of taurolidine in low, medium, and high molecular weight concentration, high molecular weight material of hyaluronic acid, you get appreciable decrease in the amount of living bacteria. But in the high molecular weight version of that hydrogel containing 3% taurolidine, everything was eliminated. So another successful outcome in a mature biofilm, which is a very difficult thing to deal with. And like Paul mentioned, in clinical practice, it's not a great thing. The other thing, it turns out that any implanted device, mostly those things you see from Boston Scientific, like pacemakers, et cetera, they will all form biofilms sometime or another, and they have issues with products that they make to create biofilms. In fact, they're having problems today with their nerve guides. Even those things are getting biofilms. So we created 8 non-woven mesh products as well. And we integrated taurolidine into meshes using electrospinning. Now we wanted to test this against the similar microorganisms we used before. But notice one thing, I was able to get 21 to 59% by weight of taurolidine in these fiber constructs. That's a heck of a lot of drug, and probably overkill. But we did bacteria kill studies, again in Professor Schultz's lab, with suspended cultures of Pseudomonas and MRSA. Okay, here are the concentrations of the living bacteria. When I introduced taurolidine in the mix and tested it against all of these materials that were created, all these bars, by the way, represent different resorbable polymer constructs. So there's 8 different types that I tested. They all degrade at different rates. So these things are just different types of polymers that degrade at different rates. You'll see some of these have some influence on bacteria. That's because a lot of them release acids when they degrade. As a matter of fact, when you get a vicryl suture or a monocryl suture, 2 of the things that occur immediately is inflammation. And once the acid degrades from the polymer construct, it actually causes irritation at the site. So that's a normal thing to see when you use sutures of vicryl and monocryl. It's not unusual. But that acid environment also is able to kill some bacteria. Well, what happens when I introduce taurolidine to this? Well, we got a big problem. Kills everything. Can't even make a bar there for any of these. So very successful out of the meshes. Okay, so now what about incorporation of taurolidine in medical devices? Well, I'm thinking of the 510(k) pathway in the US. We didn't really mention CE mark. That's a very good process for this as well. In fact, we might go to CE route before we do 510(k). But I want to list 510(k) because in the US, if you have a predicate and you develop a concept that's identical to the way the predicate works, you can use that to ask for approval of your product. And I've done this many times at Ethicon. When I was there for a little over 4 years, we launched 36 products through 510(k) process. So I know how to do it. So in terms of the use of predicate devices, the indication for use really cannot be expanded beyond the predicate referenced. All of the CorMedix concepts we created have predicates identified. We know which ones we're going to use. The next step is to prove that the device is substantially equivalent. That's why we're doing the in vitro studies. That's why we're doing some animal trials. The reason we want to keep the clinical study in our pocket at Hackensack Medical is in case the FDA says it'd be nice to see some clinical data too. So Paul and I want to be prepared for that. And it's actually good for our product to do that. So after a 510K application is submitted, let's say the FDA determines that device is not substantially equivalent to the predicate. They'll say, you know, DeLuccio, you think you've got a predicate, but we don't think so. Well, if that's the case, we also can go under what's called the de novo process. And that's another way to convince FDA that what you've got is safe. If it's integrated into the product, there should not be an issue using it. Another thing in terms of planned milestones for the pipeline, we're doing the proof of concept animal modeling during the third quarter and fourth quarter of this year for the antimicrobial sutures, the nonwoven meshes, and the hydrogels. Our development strategy is to continue to advance medical device programs through animal proof of concept. We want to prepare the data for potential 510K submission, but in parallel, we can also do a CE mark application. There's a very high likelihood we can get that approved because Neutrolin's already approved in Europe, so there is experience with the product there. So, we also want to prepare for potential approval and commercialization in the US, and like I said before, seek CE marking for all the taurolidine-based products. Now, in terms of what is the market for all this, Paul already let you know that the worldwide market for sutures, all of them, is $3.9 billion. And let me tell you, the amount of integration you get for active devices in these areas is always variable. And like I said, when I was at Ethicon, they never told us how much they sold with the antimicrobial, even though we asked constantly. They said, it's none of your business. So anyway, in terms of hydrogels, the market is up to $1.9 billion worldwide for burns, decubitus foot ulcers, and osteoarthritis. For meshes, $2 billion opportunity worldwide for hernia repair, burns, wounds, and reconstructive surgery. All of these numbers come out of market research reports that have been published. Okay, so we're ready for Q&A.
Thank you very much. You know, I can tell you, I'm sitting in my seat, I may not show it, but as a surgeon who struggles, as Dr. Lawrence does, every time you implant something, you're worried, how long is it going to take to dissolve? Is it going to get infected? What is the inflammation going to be? How do you pick the right suture? How do you know that a month or two from now, you know, it's not going to spit? This is really an opportunity to change the face of how surgery is practiced in certain areas. And it's something that I look forward to, obviously, participating in. It's similar, you know, to the catheter conundrum that we've had for the past 30 years, where I'm always worried, you know, is the catheter going to get infected? I'm going to be called in the middle of the night. And to have something like taurolidine, which, after our studies prove it again and again, can decrease this by 80%, 90%, however much it is, really changes the life of those people that do these things for a living. So this is really, really exciting for me. Okay, do we have some questions? Yes, go ahead.
Hi, Ram Selvaraju with H.C. Wainwright. Just three very quick questions. Firstly, it would be helpful perhaps to understand what kinds of predicate devices you expect to use here, and in particular relating to the criterion of being able to demonstrate that your device is substantially similar to the predicate device. Is the predicate device also something that includes an anti-infective impregnating agent or not? And if not, how do you demonstrate substantial equivalence or similarity? The second question relates to strategically how you intend to penetrate these markets, because obviously these are very large markets. And then the third question is very simply, I guess, maybe for Dr. Lawrence. Maybe you could comment on the potential applicability of taurolidine in the surgical site infection setting with things that are designed to stop bleeding. Is that a potential scenario here in which you could envision the use of taurolidine, or should we be thinking about those as two separate issues, never the twain shall meet? Thank you.
Hello? Okay. As far as predicates are concerned, I'll bring up sutures for now because I could get into a long oratory about the others. But Ethicon went through the 510K process for the approval of all their plus sutures, Vicryl, Monocryl, PDS Plus. I can use the PDS Plus application they sent to FDA that did get a K number, and all I have to do is look at what they supplied to FDA, show them my data, make it correlate to what they did, and tell them I've got a product that's equivalent to that one in terms of the way it performs. Because they had triclosan in their suture, I have this antimicrobial. It works as good or better than the one that was out there before. That's all I have to tell FDA. Okay? You got any more questions relevant to that? The same thing holds true for the hydrogels. We do have predicate products identified. Some of the incumbents had things like chlorhexidine digluconate. Some had silver. There's a host of others out there. Professor Schultz could probably read off about 100 of them if he had to, but there's plenty of those to use. There's actually more of those than there are the suture predicates. So, any other questions relevant to that?
So, to be clear,
Yes.
as long as you are no worse than the predicate device, you don't need to be
You have
exactly
to
the
be-
same, but as long as you're no worse
You
than the
just
predicate
have to be
device.
substantially equivalent. Think of those two words. That doesn't mean identical. So, it's a good legal term, believe me.
If I can just briefly tell you a very quick story, how actually the FDA, the 510K process, I'm very familiar with it because just even when I mentioned the LED mask that we took through the 510K process, they're actually pretty flexible, the FDA. So the 510K process, we based our mask, LED mask, on an office device that has LEDs built into it, and you would have to go into the office, doctor's office, and sit in front of that panel for about 15, 20 minutes. That's how broad it is. So it doesn't have to be exactly the same device, but it has to do pretty much the same thing. We already know, actually, by the FDA, remember you actually sent me those slides, that the FDA already determined that any suture will be approved through the 510K process. So in our mind, that's not an issue. As far as your question about devices to stop bleeding, which is really cellulose, I don't see any reason that terulidine cannot be mixed in with it and used at the same time. Bob, correct me if you think I'm wrong, but there's absolutely no reason in my mind that you cannot infuse terulidine into a cellulose.
Yeah, I can add to that. I use cellulose all the time. One of the fears, even though cellulose does have some antimicrobial properties, is that I don't want to leave a foreign body in. But if I had the security of some of those bars there that basically say nothing's going to grow in it until it dissolves, that's really a game changer. Yes, go ahead.
What is the longevity of the, my name is Reddy from Fort Worth, Dallas, Texas. What is the longevity of the terulidine? How long it will stay in the body?
Terulidine is metabolized as it's released very quickly. Okay, so if you put it in something that leaches out, as it leaches out, the body, metabolizes it. The nice thing about terulidine, it was discovered in the mid-'80s by Geislicht in Switzerland, and we live in a very different regulatory environment now, but they actually infused grams of terulidine into patients with sepsis, grams of terulidine into patients with peritonitis, into the open peritoneal cavity. No side effects. Gets metabolized real quickly. So the nice thing about it, and that's how, conceptually it is so useful in terms of catheters, for example, and now these type of things, where it has very high concentrations where you want it, either when it's sequestered in a suture or when it's in the catheter lumen. But as soon as it either leaches out inadvertently or by mistake, if you infuse it because the catheter is thrombosed and you can't get it out, so you have to infuse it into the body, within minutes it's gone. So it's a biodegradable bleach. Think of it that way.
Okay, good.
Thanks.
Thank you.
Yeah, go ahead.
So Prabir Roy Chaudhary from the University of Arizona. So my question was, I mean, is there a niche for many of these products, for example, in areas where sterility is difficult, whether it's in the field or whether it's in parts of the world where the process of care for asepsis is more complex? We often use new technologies to address deficiencies that we have potentially in the process of care. So I'd be very
Right.
interested in your thoughts.
I would think so. Put it this way, I'm still learning. I'm preparing, for my role here today. I got to learn a lot about triclosan. The problem with triclosan, even though it's in toothpaste, it's in hand washes, is that they've actually done blood tests on healthcare workers, and they find that people in the OR have high levels of triclosan, and that's one of the reasons why, it's undergoing intense scrutiny. Now, again, this is not about, hey, let's kill triclosan, today. But we're using that to highlight initial attempts, which have been relatively successful, an amazing awareness of the current unmet need for such products, and the fact that taurolidine, because of its metabolism, because it basically degrades to taurine, regular amino acid, and some other stuff, it's almost a natural antimicrobial. Okay, it's one step away from it. Obviously, the body doesn't manufacture it de novo, but once it's in the body, it acts very much like a natural compound where the body just takes care of it. So I agree with you. The sky's the limit here, and we'll continue looking at these things.
Yeah, and I want to bring one thing up, Prabir. There is another application we looked at. Bruce, myself, and Anthony went after a DoD application where in the field there's difficulty doing anything with sterility. So we were coming up with a device that would use UV to first attack an area where a wound would occur in the field, follow that up by a catheter setup would allow you to use taurolidine so that these guys can get IVs in the field before they end up in the hospital. So that's one thing we looked at. We also put in a patent application to cover that as well. So that's an example of something you can use in the field. And it doesn't only have to be military, it could be trauma on a highway. These guys who do work on the highways for these aggressive accidents that occur could use products like this. So yeah, we looked at other things as well. It's not like we haven't done that. So there are certain other applications out there. Yeah, and I will want to mention one thing to clarify. I hope you all realize we do have a CE mark for Neutralin in Europe. So there is experience with taurolidine in Europe right now. Okay.
I did, late at night, I have nothing better to do, so I read everything that I can get my hands on. And actually, there is data, which is astounding to me, that did you know that the length of stay in a hospital-acquired surgical site infection, the average increased length of stay is 30 days. Now, imagine how much money that costs the government, the health insurance, right? So to me, this is astounding. If you can bring that down even by 10%, that's a huge saving. And imagine what the insurance companies will be thinking along the same term. So 30-day increase in hospital stay with an infection. I found that astounding.
One of the things about drugs in general and why antibiotics have been the orphan of large pharma is that if you put someone on an antihypertensive medication, you take it every day forever, okay? Antibiotics is as needed only, okay? One of the benefits of an antimicrobial concept is that everybody gets it because you don't know who it's most important in, okay? So it has a much wider, potential base. So if you look at surgical site infections, 2% is where we are now. But potentially, and this is, again, the quote directly from the World Health Organization, they're literally recommending that everybody should have an antimicrobial suture, even though society is simply not ready for it. So, conceptually, if an SSI is a disaster, and under certain circumstances, in many circumstances, it won't be preventable even with sutures because of the nature of the wound itself, you still have a tremendous potential to avoid many cases. So, Prabir, to answer your question, we're just beginning to figure out the potential applications here. But if you look at wounds, for example, especially well-established wounds or burn victims where, it may take a month to prepare the wound for a skin graft until it granulates. Part of the problem is established biofilm. So if you have something that not only prevents infection as a microbial, but can very quickly get into the biofilm and displace it, okay? You're talking about potential changes in many different chronic wound scenarios. Now, will this pan out? Who knows? We got a lot of research to do. But all the data that we see here, both the conceptual basis and the data that we see, point to a very promising beachhead into something which can be very impactful.
On the antibacterials, I know it's a huge problem, as you mentioned, on implanted devices. And could you give some assessment as to other companies that you know of that are working on this problem in terms of what competition might be on the horizon and where you may stand in the race? And I know this is very premature, but I'm just curious about how pricing might be when you weave this into, say a pacemaker or something. Because it would seem if you're getting a 0%, if you're killing all the bacteria, it would seem, if I'm the FDA, I would want to mandate, I wouldn't want to pay for devices that don't have, it would make no sense, because you're talking about increased risks of infection, long hospital stays, as you just mentioned, et cetera. Thank you.
Okay. I'm actually not worried about competition because the devices that are out there right now with the chlorhexidines and the silvers, et cetera, they all have problems. And all those companies are looking for alternatives. Because of the issues that exist with the current products, they want to have an alternative. In fact, when I went to the Jacob Javits Center to a medical device trade show, I told folks at 3M, they have a Tegaderm with chlorhexidine digluconate. I said, I have not a new antimicrobial, I have an antimicrobial we believe would be effective in your product. They said, we're getting our director of research and development to get in touch with you. We do want to do a collaboration with you. So that's an example of a company that's well established in the community that asking me to help them. So that gives you an idea. Go ahead, you got another question? I'm actually not concerned about the pricing. We almost can charge what we want for this. I don't want to go crazy. I want to be as competitive to what already exists. And I think we can overcome what's already there. So I'm not worried about the pricing per se. And in fact, if it's more effective than what they've already got, they'll be willing to pay a premium for our product. So that's just my feeling about this. I mean, we can make bets about this, but that's my feeling about it.
So, Bob, a follow-up to your statement and a question to Dr. Lawrence. What is the importance that is put on wound healing, in preventing infections with wounds, especially in the general area, as Gary's referring to, in terms of preventing length of stay in the hospital, but also when you're doing aesthetic procedures with patients? What type of importance do you as the aesthetic surgeon and does the patient place on having a wound heal quickly and eliminating infections? And is that a plus for you? And do you, in the self-pay market as well as the insured market, what are your thoughts about, a product in terms of reimbursement?
Well, I'm glad you mentioned that, because when it comes to aesthetic surgery, it's sort of I almost think of aesthetic surgery as being non-surgical in a certain way, because I really have no room for error, right? When you do open heart surgery and you take that patient back to the OR and infection and patient eventually dies, that's acceptable because it's just what happens. In the aesthetic market, it's not acceptable. People pay a lot of money and expect a perfect result each time. So the incidence of infection in what I do is small, relatively small, but it can be devastating. So it's very, very important. The other thing is, as far as bariatric surgery, much more common, huge flaps. We raise huge flaps when I do body lifts and abdominoplasties. Incidence of infection goes higher. So when you do body contouring, really the real number is 30% complication rate, and most of that is infection. So if I have a suture that can minimize that 30%, I'm using it. I better be using it, otherwise I'm not within what the community is doing it. So to your point, it's hugely important in what I do in the self-pay market especially.
Just want to add one little thing in terms of just devices. We're talking about sutures that have either interstices if they're woven, or you can co-extrude it into sutures that dissolve over time, okay? Had to have tyrolidine potentially on a stainless steel or titanium surface is a whole nother problem. So, you know, we have enough to deal with to keep our plates full for a long time, but you know, I want to be honest with you regarding what the big scope of this is. You know, the scope at this point, as far as we can envision, is not putting tyrolidine on every surface that exists in the body. You know, we still have more work to do before we can envision that. But as relates to implantable devices, or for example, a suture that you put in a bariatric surgery or cosmetic surgery, that all you need is for it to get infected, and it's like, oh my God, this stitch is going to sit there for a month. It's going to rise to the surface. I'm going to have to open up the wound. I'm going to have to fish it out. If you have a suture that basically says, I simply don't get infected, okay, that really changes things, even if the underlying wound potentially is a wound that gets infected itself. Because part of the problem surgically is you have a wound that's not that good, okay, the wound eventually takes care of itself, but then you have all these little stitches that have dotted it, and those aren't alive, so to speak. Now, again, we're projecting forward. We really don't know how a lot of these things will work in real life. All I can tell you is this is real exciting. That's all I can tell you. And based on the success of our competitors in capturing 10, 15, 20% of the market and the needs, you know, of the cosmetic market, bariatric market, and others, you know, really, this is something that we're pushing really full speed ahead. Okay. Well, thank you very much. The next segment here is talking about anti-tumor agents. Tyrolidine has been shown to have anti-tumor properties, and nothing clinical yet. In cell cultures, it's been shown to have a synergistic effect with some pediatric cancer regimens. The challenge, as we alluded to, is that if you take tyrolidine and you infuse it intravenously, it gets metabolized. So somehow, as with tumor agents, you really want to have localized delivery, high concentrations, and as such, the delivery system becomes very important, and those are one of the things that we're working on. You can't do this in a vacuum, and Tony and others will talk about our collaboration with the Poetic Group, which is Pediatric Oncology Experimental Therapeutics Investigators Consortium. That's a fancy word. I'd like to introduce the next group of speakers. Actually, just Dr. O'Neill, and Tony will also come. Dr. Neal is a assistant professor of pediatrics at Harvard Medical School. She's a pediatrician as well as a pediatric oncologist with a pedigree education at Yale, Einstein, Boston Children's Hospital, and the Dana-Farber Cancer Institute. Her clinical experience is in the care of children with solid tumors, and her research focuses on novel imaging techniques of these tumors. She'll highlight the great need for cancer treatment innovation. We thank her, of course, for her work on behalf of these unfortunate children and for her participation today. Dr. O'Neill?
Hi, everyone. It's a pleasure to be here today. I wanted to give you an overview of pediatric neuroblastoma, particularly in light of the fact that it's one of our most common tumors, the most common solid tumor in pediatrics, for which there's a great need clinically to improve outcomes. So briefly, we'll talk a little bit about the pathophysiology of the disease, the staging and risk stratification as it pertains to outcomes essentially in these children, and the treatment approach and where we really need more help, and why a drug like terulidine, with its promising results thus far, could really be helpful in this regard. So neuroblastoma is essentially a small round blue cell tumor. It arises from the neural crust cell, so it's a neural origin tumor, which means it arises either from the adrenal glands right above the kidneys or the parasympathetic nervous chains right on either side of the vertebral column. So these tumors can really arise in many locations within the body. They tend to encase really crucial structures, and they have vast metastatic capabilities, so disease can be quite widespread at the time of diagnosis. The hallmark of these tumors is that they secrete catecholamines, essentially hormones, that can cause high blood pressure and a number of other symptoms in these children. This is just taken from a New England Journal of Medicine article in 2010. Essentially shows you the vast array of locations where you can see these tumors. They can occur, as I mentioned, in the paraspinal region, so they can cause spinal cord compression and paralysis. Many children actually present in this fashion. They can involve the organs, predominantly the liver, causing massive abdominal distension and respiratory compromise. They can involve the bones and the bone marrow, and of course can impact many of the neural structures, as I mentioned, causing neurologic symptoms other than spinal cord compression. Most children present with extreme irritability, likely due to pain. They're often quite young, so they can't necessarily tell us why they're in pain, often with a large abdominal mass or distension. Raccoon eyes are kind of a pathognomonic feature in the sense that these children have invasive disease in all the bony orbits of the face, so they present much as this child does here. A lot of the times the children are actually worked up for child abuse before the actual tumor is diagnosed. But certainly there's a whole array of other symptoms. This disease can really masquerade as so many things. It's traditionally diagnosed in children less than 5 years of age. This is taken from the SEER database, and it essentially shows you the decline after a certain age point. The median age is really around 3 years of age. There are 700 cases a year. As I mentioned, it's the most common solid tumor in children and one of the most devastating. And there is a small cohort of children diagnosed less than 18 months of age. These tend to do better than the others, and disease can actually spontaneously regress, but they're a very small minority of cases. Disease is staged in a surgical fashion or by imaging, and really the point that I want to make here is, of course, like any other disease, the higher the stage, the worse the prognosis. And while localized tumors can be a low stage, a stage 1, and perilocalized tumors, a stage 2, once you get to a stage 4, which is 50% or more of children, your outcomes plummet drastically despite really intensive multimodality care. More recently, we've adopted a different staging algorithm that looks at imaging-defined risk characteristics. So it essentially tells us how much these tumors wrap around really vital structures, which impinge upon our ability to surgically resect them. And so in addition to chemotherapy and targeted drugs, surgery is so important for the cure of these kids that when we have tumors that we can't resect, it vastly impacts outcome as well. There are a lot of points that come into risk stratification for these tumors, the most important of which is NMIC amplification or MIC-N amplification. It goes by either term. This is a gene on chromosome 2, greater than 10 copies of which portends a very poor prognosis. So you can see on the survival curve on the right, these are children less than 12 months of age. They often do better, but if the tumor harbors NMIC amplification, you can see that their survival plummets. They do much worse. And so this is a risk stratification factor that is quite important to us. And if a child's tumor comes back NMIC amplified, we know that we have a much bigger struggle on our hands. In addition to stage, as I mentioned, so extent of disease, presence of MIC-N amplification, and child age, those are really the most important factors that allow us to risk stratify these patients. While patients can fall into a low risk or an intermediate risk categorization, the high risk category, as I mentioned, greater than 50% of patients are the ones for whom we really need more therapies. Low or intermediate risk patients can essentially get by with observation or some degree of chemotherapy. They're a smaller cohort of patients. They have about an 85% and upward survival, so not bad, but the patients that do relapse are often incurable. And when we look at the high-risk patients, I actually included the schematic on the left, not so that you can go through all the details, but to show you the vast amount of therapy these children need. We're talking upwards of a year of treatment with inclusion of every modality I listed on the right-hand side. So they need very intensive chemotherapy, surgery of their primary tumor, they undergo autologous stem cell transplants. So they're given back their own stem cells such that we can give them even higher doses of chemo. They get radiated, they receive maturation agents, and most recently we've given them immune therapy as an adjunct to their care. So very multifaceted treatment, and yet these patients still don't do well. This is one of the most recent studies taken out of the New England Journal that showed any improvement in overall outcome in decades. These are patients that were randomized to the whole slew of agents that I just showed you with and without immune therapy. Immune therapy improves survival in these patients to up to about 66%, but even then, that's the best we can do. And while there are new agents on the horizon, something like terulidine, if given in concert with some of the agents we already give, could improve outcomes for 50% or more of patients. It's certainly something that would be exceptional for this patient cohort. And finally, patients with relapse disease really have a dismal outcome. There are nuclear medicine therapies we can give. There are experimental agents that we try alone or in combination. But at the end of the day, those patients too are incurable. So if we can come up with better drugs like terulidine, if we can study them in concert with agents we already use or in modalities that we already attempt for these patients, I think we would be much better off. And I know that Tony's going to speak about Poetic next. I think coming from the perspective I do, where these are rare diseases and we have to work in a consortium setting, the opportunity to incorporate new agents for diseases like this is really important in the work we do. So, thank you.
Thank you very much, Allison, and thank you for coming down from Boston today for the presentation. Very important disease process and important presentation. So I wanted to discuss our collaboration with Poetic, which we instituted, and we had been looking for a development group for neuroblastoma. And as Gary had mentioned, I had worked for 7 years at Sloan Kettering as a nephrologist, mostly putting in bedside catheters and ordering TPN, but at the same token, knowing a lot of the oncologists there, I reached out to the pediatric oncology group and essentially was heartened to know that they were very interested in terulidine and neuroblastoma. It wasn't something I had to push, and they were willing to help us to progress research in this area, preclinical research, which we needed to accompany our orphan drug effort, advancing it. And they were able to come up with a program which we negotiated with them, a very exciting program and a very economically efficient program. So without further ado, I'm going to describe what is Poetic. And the reason why there's a consortium, one might say, why not just go to MD Anderson or MSKCC or any of the other institutions here, because these are rare pediatric cancers. And they're not very commonly found. So in order to do large clinical studies, Tanya Trippett, who's a physician and head of pediatric oncology research at Memorial, developed this consortium of institutions, which I'm going to visit up in Alberta later this month. They include MD Anderson, Memorial, University of Texas, San Antonio, Phoenix Children's Hospital, Alberta Children's Hospital, the Arnold Palmer Children's Hospital, Penn State Hershey, Arkansas Children's Hospital, and Lucille Packard Hospital at Stanford University Medical Center. Okay. What are the abilities of Poetic to help advance our programs? An example is that Alberta Children's Hospital, Dr. Aru Narendran, the professor and director of that preclinical program, does a large amount of preclinical research for Roche Genentech. It's one of their major collaborators. So they're experienced at developing drugs. They have academic as well as experience in drug development. So far, they have conducted cell line studies and they have started animal studies for us, which is something that is essential to our aim at developing the drug. Essentially, they're looking for best targets. They're also looking to more perfectly characterize the mechanism of action of terulidine in cancer. So They're characterizing its mechanism, and they're looking at targets such as neuroblastoma and osteosarcoma, and they're looking for the best agents to combine taurolidine with. We mentioned vincristine, but they're also looking at cisplatinum and taxanes like Taxol and Taxotere. Okay, to concentrate the particle, as Bob mentioned, it's a polymeric delivery system. It's biodegradable as well. So it delivers the package, the drug, to the cancer cell, and then it dissolves, the basket dissolves. It's a slow, sustained release, and it is preferentially taken up by tumors, as we see in the cell line data we've already developed. Patents have been filed thanks to Bob and our patent attorneys in the EU, US, and Japan covering nanoparticles, and also the ability to deliver taurolidine as one therapy versus a combination therapy, as I just discussed, has been worked upon. Here's some examples of the use of nanoparticles, nanotechnology, to deliver paclitaxel and the inducement of apoptosis or programmed cell death in breast cancer cell lines. So it's not just relegated to neuroblastoma cell lines. That's our first target, but it also seems to have an effect on cell lines in breast cancer in combination with a commonly used chemotherapeutic agent like paclitaxel or Taxol. Okay. This is some example of, if you look here, we're not talking about patient survival. I did a presentation once, and so some person commented that the survival was essentially not good. It was going down, and I had to remind the audience we're talking about preclinical data now. So you're looking at concentration, and you're looking at survival of cells, okay? So nanoparticles were tested in a resistant cell line, neuroblastoma resistant cell line, NMYC, which was essentially highlighted by Dr. O'Neill in her presentation. And if you look here, it shows that there's a significant decrease in cell survival. If you look at the IC50, which is a marker of cell death, you see that if you combine the agent, you see increase in cell death. So it kills the cells in an effective manner, the cancer cells, that is. Vincristine nanoparticles against neuroblastoma cell lines. We also looked at, or the scientists who conducted this data for us looked at concentrating the vincristine nanoparticles, and you see also a nice decline in the number of cells. And then when you combine taurolidine and vincristine together versus vincristine alone, so this vincristine, taurolidine, and vincristine, and the percent survival of the cells, you see here that there's a big reduction when you, in combination, used vincristine and taurolidine at the 0.75 nanomole vincristine and 35 micromole taurolidine concentrations. And if you look at the neuroblastoma cell lines in NMYC non-amplified cell lines, neuroblastoma cell lines, these are cell lines that are from Memorial Sloan Kettering Cancer Center, SK and SH. You see again a dramatic reduction in the cancer cells, so it's killing the cells. These were tested in another cell line, the BETM1 cell line, which is an NCI cell line, which also showed a significant reduction in cancer cells. Again, it's not restricted to neuroblastoma. This is cells that were tested which were non-small cell lung cancer cells, and it seems that taurolidine nanoparticles were also effective at reducing or killing cells. Of course, this data has to be replicated, which we're starting the process now in animals. That's underway, but this is encouraging cell line data, preclinical. Taurolidine, exciting prospect for pediatric oncologists. Again, there was a sense of collaborative excitement with Poetic. It wasn't that we said, okay, we would like you to do these testing, and they were ambivalent. They were encouraging us to move forward with the program and have been very supportive of those efforts. We are also aware that it's important to have proprietary patents filed. Bob has done that in general terms, and we're working with a proprietary cell line. CorMedix expects to have the opportunity to license the technology. So what we want to do is finish the preclinical. We were given a recipe of what we need to do, which Poetic is fulfilling. We have other advisors, consultants helping fulfill the steps that we need to complete. And then at some point in time, once we have animal proof of concept, we would look to leverage that information, as Koso said, to then potentially do some type of partnering in oncology, which is obviously a very advanced and complex area to navigate. But we've gotten off to a good start, a good foundation, and we're very happy to have this ability to progress with such good advisors and good groups of people like Poetic and Dr. O'Neill.
Thank you, Tony. Thank you, Dr. Allison. So this concludes the second part, the anti-tumor effort. Any questions? Yeah, go ahead.
Just very briefly, could you remind us what the nature of the nanoparticle delivery system is that you're using here, and whether you would make any modifications to it going forward, and if so, for what reason?
It's a terpolymer. You know what that is? It's made of 4 different building blocks. 2 of them are PEG-related, polyethylene glycol. 1 is lactide-related. The other is glycolic acid. When you make different mixtures of those things, they degrade at specific rates. Lactide tends to degrade slowly, glycolide pretty quickly. Polyethylene glycol just lets things stay. It really does not degrade all that much, but it does get excreted in urine. So not an issue if you use the right molecular weight of it. So the 4 segments are put together in a specific pattern. It's called a random terpolymer, which means you put the components together, they assemble the way they want. In terms of making other types of nanoparticles, the answer is yes, you can make umpteen thousand of them if you want. We decided to just test one and see how well it works. So far, it delivers the drug over a 3-week period in vitro. To us, that seems to be okay, but if we need to modify it, it's not a big deal. Process is simple enough that it's not difficult to do. Okay?
Sounds good to me. And if you can repeat that, you get a prize. Okay, any other questions? Yeah, go ahead.
So this is sort of more, and this may be my naivety, and it's more of a scientific question. Right. So some of the actions that you've described for taurolidine, which are clearly good in terms of its anti-cancer, anti-proliferative effects, could they be a negative when you're putting it into sutures for wound healing? And that may or may not be the case, because other drugs like paclitaxel, for example, that probably hasn't happened, but I'd be interested in your thoughts.
Yeah, it's a good question. I haven't thought of it, but I can think on my feet. You know, I can try to. You know, when you put sutures in, you know, you're just sort of holding the tissue together, and I wonder if the zone of inhibition would extend that far into the wound, and clearly you wouldn't want that to happen. But what I would think is you'd be able to dial it down so that the antimicrobial effects are not extending into the wound, because that's not really what you want. I don't see sutures in general, and I'd like to hear Dr. Lawrence's opinion on this, you know, as, hey, we want to give sort of antimicrobial to bathe the wound so that it should heal better, you know, and not get infected. I believe that this concept is more, we don't want a nidus of infection to take place in the suture while the wound is doing its thing, because the suture doesn't have macrophages to get into it, so to speak. And once the suture have a foreign body localized around it, you know, that's sort of the body can't do its normal things. So I would think we would want to dial it down so that sutures don't get infected, they don't carry on the infection, and they just sort of are taken out of the equation for the body to do its thing as it comes to wound healing. That's the concept. I'd like to hear your perspective.
I agree with the concept. The other thing that I'm thinking is, occasionally not occasionally, but not infrequently, I come across hypertrophic scars and keloids.
Right.
On breast reduction patients or abdominoplasty patients. And the question is, would the anti-inflammatory property of a pteryglidine suture cut the incidence of that? Because that's a huge problem.
Right.
So
No, absolutely. I mean, around any suture, you have inflammation, and the more absorbable a suture is, the more inflammation you have. That's a basic concept. In other words, if you put a suture in that lasts forever, proline, almost no inflammation. You put a suture in like chromic catgut, okay, which takes 2 weeks. There's a tremendous amount of inflammation. You put in Vicryl, which may last 4 weeks to 6 weeks, the inflammation is less. So the body sort of attacks those things that it can get rid of, and it sort of sits back for those things that it can't get rid of conceptually. So if we can start playing with that and basically saying we want to decrease the inflammation, okay, even for those things that you get rid of quickly, okay, many, many different possibilities. Bottom line is again, I can only share my excitement to be able to potentially modify these things hitherto that we could modify, because suture is a suture and that's it. Go ahead. Go ahead.
I'm sort of answering my own question, but I will share some of our experience. So we did a lot of work with paclitaxel, which clearly is a very potent anti-proliferative agent, and this has obviously been shown clinically as well. But in our pig work, for example, when we put wraps that had been impregnated with paclitaxel around the suture of an arteriovenous graft, for example, we actually got no breakdown at all. So we were concerned that there would be breakdown, but we really had no such problems. So I'll just add that in as well.
Right.
If I can make a comment, we will have, Bob and I will have the answer to your question in about 2 months. So call me in 2 months.
That long? Can we?
No, I'm sorry. I have to make a living on the side too, right? But we will have that answer to your point, because that's exactly what we're doing in the pig model, right? And that's going to be the first step. And then our plan is to approach the FDA, and then, but of course, already start the clinical. But that's going to be answered in the animal model.
And
also suggest, even though I'm not a surgeon, that having some anti-inflammatory effects in the setting of trauma, etc., may actually, just as you suggested, in terms of scarring and all, could actually be a positive.
And one of the reasons that we are using Hackensack is that they have an incredible lab, and we have so many different matrices that we are measuring the wound quality. So it's not only going to be a visual, but it's a photographic documentation, and it's a real valid way of looking at wound generation of a wound.
Right. Prabir, as you know, but I'm saying this for the benefit of those that may not, there's so many different phases in wound healing, okay? So that, you have the acute phase where it basically sits around, doesn't do much. And then, the body puts in, all the cellular healing, cascades. And the point is, we simply don't know, and we can't, of course, balance at this point when we want these effects to be maximized. For example, the delivery system. So is this something that you want to sort of lay low for the first week and then start having pteryglidine, come out? Or is it something that you want to give your dose initially, because that's perhaps where the infection takes place, and then you want to come back? So there's so many things, and that are happening, that are dynamic, and that may be dependent on timing, or it may be just sort of, dumb antimicrobial, just leave it there, you're fine. Who knows?
And in a way, I'm sort of answering my question. But many years ago, I was very interested in TGF beta, and there's a Nature paper from maybe 15 or 20 years ago where they injected TGF they injected an antibody to TGF beta into a wound. Scar formation decreased, but the tensile strength of the scar actually improved. So, actually, that just comes back to me. So that may actually argue for a benefit of something that's anti-inflammatory, paradoxically.
Right. Okay. So, Neutralin antimicrobial solution, this is sort of our first love, okay, for this company for sure. And why Neutralin, okay? And again, I just want to give an overview. I'll leave the details, of course, to the presenters. Catheter infection is a huge problem, okay? With great morbidity and billions in healthcare costs, we have addressed in patients on dialysis, but the truth is any chronic catheter, certainly when it comes to cancer patients, long-term patients in the ICU, it's all, you know, the same problem. Just to give you a sense of how big the problem is, and not only in terms of numbers, which seem to be going down a little bit, okay, but just from the impact that a catheter has on an individual's life and on their propensity to get infections. In dialysis, 20% of patients have a catheter, approximate numbers, yet these patients have 70% of all infections. This is data just released from 6,000 dialysis units. It's mandated reporting today. So the 2014 data were collected, just released by the CDC, an article of literally a week ago. And so we have the numbers. Now, there may be some underreporting, but you know, you have a situation where if somebody is condemned to a catheter, and you may use that word, you're increasing their infection rate from 6 to 10 times as compared to if they had a fistula graft. Now, little caveat, it's possible that the sicker patients get the catheters, so that's why they have more infections. But it's incontrovertible. 85% of this increase in infection is directly attributed to the catheter. Now, the catheter, it's a piece of plastic. Since when do pieces of plastic get infected? It's inanimate. But we understand why, because of biofilm, okay, and basically adherence of catheters like moss to a rock. They've developed all type of ways of adhering and staying there, and Neutralin has shown great promise, great basic science, great clinical results in Europe, which show essentially that it deals with this problem and almost eliminates it. So the question would also be, well, okay, we have this new medication or drug or antibacterial. Why not just treat them with antibiotics or do prophylaxis with antibiotics? Well, we've addressed that before. I don't want anybody to think that this is not novel therapy. This is really groundbreaking novel therapy in that if you take antibiotics and you instill it in the catheter, which at times is done if you want to treat the catheter, what's going to happen is you're going to get more and more resistant bugs. So that's really a no-no. Now, the medical need for antimicrobial lock solution that does not develop resistance is another challenge that we're charged with, and we've essentially accomplished that thus far. There have been no reports of resistance. So our taurolidine-based lock solution has heparin for thrombosis, citrate for the pH, but essentially the core component is the taurolidine. And again, as I mentioned before, taurolidine is not toxic in the bloodstream. And I say this to compare it, for example, to citrate, where there are various competitive products where they'll use either a little bit of citrate, which essentially mimics heparin in terms of the anticoagulation, but to get antimicrobial effects from citrate, you have to dial it up to approximately 40%. And then if by mistake the patient's nurse or someone who doesn't deal with these patients says, well, if I can't withdraw it, I'm going to inject it, it has caused cardiac arrest, secondary hypocalcemia, et cetera. So the bottom line is that, we'll talk about this a little bit more, but the essence is that this is really an unmet need, and taurolidine brings a lot of benefits to this. So we're going to have two speakers that I need to introduce. The first is Dr. Greg Schultz. Dr. Schultz is a biochemist with extensive experience in cell biology. He's a professor at the University of Florida, where he established the Institute of Wound Research to study molecular and cellular regulation of healing, focusing on the roles of growth factors, cytokines, and proteases in normal and chronic wound healing. He has published more than 250 research papers, chapters, and review articles, which have been cited in excess of 6,000 times. It's pretty amazing. He has over a dozen patents in the area of wound healing. He's an accomplished researcher, having worked on dozens of projects sponsored by the National Institutes of Health and pharmaceutical companies. He has been recognized so many times and has too many awards for me to mention, and we're glad that he is here today. Now, just the amount of studies that he has done or is in the process would keep someone busy for half a lifetime. So we hope you continue with us.
Thank you, Dr. Gilfish. Well, good morning, everyone, and it is a pleasure to have an opportunity to tell you today about some of the experiments that we've been doing and how this, I think, fits into the overall strategy and development that CorMedix has. Now, we've heard a lot about infections today. Obviously, neutrolidine is an antimicrobial, and I'm going to focus down a little bit more on an area that you may not be that familiar with, and that's with bacterial biofilms. So let's step one pace back and let me explain to you a little bit about what a planktonic bacteria and what a biofilm bacteria is. And again, you may well know this, but in case you don't, it's critical for what I'm going to talk to you about. So this diagram, which comes from one of the pivotal studies that Bill Costerton published, this was in Science in about 1999, so you can see just by the time, this is a relatively new area for microbiology and especially in clinical applications. Bill was the person who coined the term biofilms, and the important concepts is these white ovals here are representing single planktonic bacteria. Planktonic bacteria, those rapidly proliferating, non-attached, single bacteria that we probably all grew when we took undergraduate microbiology lab and cultured bacteria on agar plates. Normally, our immune system, our antibodies, as well as our inflammatory cells, our phagocytic neutrophils and macrophages, are usually very effective at controlling the number of planktonic bacteria that would be present in a wound or in other parts of the body. But when planktonic bacteria initially reversibly attached to surfaces begin to proliferate, they will develop localized concentrations of those bacteria. And almost all bacteria species have the capacity to do what's called quorum sensing, which means that the bacteria have evolved systems where they secrete unique molecules that communicate their presence to other bacteria in the area. And as the name implies, when the concentration of those interbacteria signaling molecules reaches a threshold, a quorum, the receptors for those quorum molecules get triggered in the bacteria, and it dramatically changes their pattern of gene expression. So now, instead of proliferating as rapidly as they can and not being non-attached, these bacteria began to take most of their energy and they use it to synthesize and secrete what is called an exopolymeric matrix, or slime if you're a microbiologist. So this exopolymeric matrix consists predominantly of relatively unique polysaccharide chains, as well as unique proteins, and surprisingly about 20% of a lot of this mass of the exopolymeric matrix is actually free bacterial DNA. That's important because our innate immune system, our toll-like receptor system that we're all born with, is able to recognize that unique sequence within free bacterial DNA, the so-called CpG islands, as well as these unique polysaccharides that the bacteria synthesize and secrete only when they're in the biofilm phenotype. Now, as I'll show you very briefly, that when bacteria are present in this exopolymeric biofilm structure, they develop tremendous tolerance to our antibodies, to our inflammatory cells, and unfortunately also to many of the typical antiseptics and other materials that we would use to try to kill planktonic bacteria. So what happens then is this biofilm community persists even after repeated attempts by our own body to kill it, or by repeated treatments with antibiotics and many antiseptics. But because the exopolymeric matrix of the biofilm, as well as many of the exotoxins that the bacteria are secreting, it produces a tremendous inflammatory response. And that draws in more and more neutrophils and macrophages. They activate and secrete the things they normally use to engulf and kill bacteria. They generate and release reactive oxygen species, and we heard some about that earlier today. They also, importantly, secrete several different proteases, including matrix metalloproteases, or MMPs, and a serine protease called neutrophil elastase. And it is the elevated persistent concentration of these reactive oxygen species and the proteases that the neutrophils and macrophages are secreting that have off-target effects, because they're not able to kill the biofilm, but those products are able to damage the surrounding host cells. And the result of that is that we see the chronic ulceration, chronic pain, inflammation when a biofilm is present on tissues or on inert objects such as catheters. Now again, recognizing that the field began to understand the importance of bacteria in the biofilm phenotype, in 2007, DePozo and Patel published this really interesting review that listed 18 clinical pathologies that in 2007 were clearly identified as being caused by the presence of biofilms producing this chronic inflammatory state. Particularly important for CorMedix is the recognition that catheter infections, dialysis catheters, urinary stents, all of these chronic infections are really caused by the presence of bacteria in the tolerant biofilm phenotype stimulating chronic inflammation. Now, it's not just inert devices. We also know that biofilms are the main causative agent for things like chronic sinusitis, and as we heard from Dr. Lawrence, particularly in the chronic inflammation that will develop around many breast implants 2 or 3 years after the surgery has been completed. And of course, almost every orthopedic chronic infection is due to the biofilm, either on this adjacent soft tissue or actually biofilms growing on the orthopedic hardware. So the point is that biofilms are now recognized as one of the most important kinds of chronic bioburden infections within the body. So I'm going to show you just 2 slides that illustrate how difficult it is to kill bacteria when they're in the protected biofilm phenotype. So again, Bill Costerton, in a great Scientific American article, produced this result. He grew a mature biofilm on an inert surface, a plastic surface, and then treated it with dilute Dakin's solution. Dakin's is basically bleach. It's hypochlorous acid. And he treated it for 60 minutes, an hour, and you can see when they stained it with dyes that detect dead cells or live bacteria cells, the outer layer of the bacteria in the biofilm were actually killed. But importantly, the bacteria that were deeper in the biofilm matrix survived after 1 hour of exposure to bleach. Now remember, bleach will kill, at this concentration, will kill planktonic bacteria in about 15 seconds. So why was the bleach ineffective at killing these bacteria that are deeper in the tissue of the biofilm matrix? Well, it turns out that when the level of chlorine, the active agent in the bleach, was measured, it turned out that even after 90 minutes of exposure, the chlorine had only penetrated about half of the distance from the surface to the base of the structure of the biofilm using chlorine probes. And this is what's been called the reaction diffusion problem. So many of the most commonly used antiseptics are chemically reacting. That includes hypochlorous acid, things like silver. And they get consumed by reacting with the components in the outer layers of the biofilm matrix. And they just can't penetrate deeply enough into the biofilm matrix to kill all of the bacteria in the time frame and at the concentrations that are used. So, hypochlorous acid rapidly reacts with molecules that form the biofilm exopolymeric matrix, which limits its diffusion into the center of the biofilm colony. And again, that's the reaction diffusion problem. Well, what about antibiotics? Again, the effect of antibiotics on planktonic versus biofilm bacteria is phenomenally different. So Walter's group basically took Pseudomonas aeruginosa, grew them as rapidly proliferating planktonic cultures, exposed them to 50 times the minimum inhibitory concentration of tobramycin, and in about 4 hours, every planktonic bacteria is dead. That's what antibiotics do. They kill rapidly proliferating bacteria because they interfere selectively with bacteria, protein, enzymes, and membrane systems. In contrast, taking these sensitive planktonic bacteria and growing them in a mature biofilm and exposing them to 4 days of that same concentration of tobramycin that killed every planktonic bacteria in 4 hours, killed less than 1 log. So this is a status of the bacteria that was tolerant to the antibiotics. This is not genetic resistance. Resistance and tolerance are very different to microbiologists. If these tolerant biofilm bacteria are ultrasonically dispersed back into single cells and exposed to the antibiotic again, it kills them just like it did before. So this is a consequence of the bacteria in the biofilm phenotype. And one of the major reasons they develop this tolerance is because when bacteria within biofilm communities mature sufficiently, they develop metabolically inactive or dormant persister cells. So Phil Stewart at Montana State did a really clever experiment to illustrate this. He took Pseudomonas and transfected the bacteria with a plasmid that expresses green fluorescent protein only when the bacteria are metabolically active. And when he grew these colonies and looked at them with confocal microscopy, you can see that only the outer shell of this biofilm matrix is actually green. Only the outer cells are metabolically active. And for the second time, what do antibiotics kill? Only metabolically active bacteria. So that means that the outer shell of this biofilm, mature biofilm community, is sensitive to the antibiotics, but the bacteria that are in the middle of the biofilm are metabolically inactive. They're dormant persister cells that can survive exposure to the antibiotic. Again, just like with the bleach, if we ultrasonically disperse these back into single planktonic bacteria, expose them again to the antibiotic, then they'll be killed. Now, you heard from Dr. Lawrence previously a little bit about some of the work that we've been doing with them, with CorMedix, on looking at the effect of taurolidine in various polymers like sutures, in gels like hyaluronic acid, in washes like Neutralin. And again, just to remind you, when we examine the capacity or assess the capacity of fibers of sutures or polymers containing taurolidine in different formulations to kill planktonic bacteria, we find that there are very effective concentrations of taurolidine in different polymers that can generate killing of planktonic bacteria. Now here, just to remind you, high numbers are good because we're looking at the diameter of the zone of inhibition. We want big zones. We want it to kill effectively. And again, whether it's in a caprolactone or a dioxone type of polymer at different concentrations, you can see that the dioxanone a polymer with 10% pterolidine basically had tremendous killing of the planktonic bacteria. Now remember, what we want to do is ideally prevent biofilms from forming, because we can kill planktonic bacteria before they develop into the mature biofilm. It's really hard once they get into that biofilm. So this is the concept of prevention of biofilm formation. Well, what happens if the bacteria have already formed a biofilm? So we've developed a laboratory model that generates a tolerant, mature bacterial biofilm using the most relevant matrix we can for skin wounds. And this is a pig skin explant model. We basically take fresh pig skin, we create partial thickness injuries, sterilize it, inoculate it with bacteria, and we grow it for anywhere from 3 to 5 days, depending on the bacteria, to develop a mature tolerant biofilm. And in fact, we can even submerge these explants in antibiotic solutions for 24 hours to kill every planktonic bacteria, and all that is left is a truly tolerant biofilm. We can then test the effect of different treatments on killing and dispersing that mature biofilm. And as Dr. DeLucchio indicated, when we look at the effects of pterolidine-containing solutions on killing these mature biofilms grown on explants, we find that many of the pterolidine solutions have very effective reductions in killing of truly tolerant mature biofilms, killing almost 6 logs to 7 logs of materials compared to the controls. In addition, as you've also heard previously, hyaluronic acid is a tremendously useful component of the extracellular matrix. It has inherent growth promoting activities, wound healing activities by itself. And again, when different concentrations of pterolidine in hyaluronic acid gel alone or with other components are tested in the mature explant model, it was totally able to completely kill the biofilms. Now, step aside for 1 second and say that we have looked at essentially every commercial dressing available for wounds. We have found one other currently available dressing that will kill mature biofilms, and that is a cadexamer iodine-containing dressing. Those are Iodosorb and Iodoflex. Nothing else comes close. We can get every silver, every PHMB, every chlorhexidine dressing. They'll reduce about 1 to 2 logs. Nothing does this except the cadexamer iodines. And it's partly because of the sustained release of the iodine from the cadexamer beads. So again, going back to the concept of a sustained release form of pterolidine in nanoparticles or in the appropriate polymer combinations makes a lot of sense as to why we were able to see these kinds of effects. Also, pterolidine, by its multiple mechanisms of killing bacteria, doesn't require the bacteria to be metabolically active. It can disrupt membranes and kill through other mechanisms that do not require the bug to be metabolically active. So in summary, what I hope I've done is indicate to you that biofilms are present in very high percentages of medical devices. I showed you the 18 examples of that containing medical devices with catheters. Chronic skin wounds, we've just published 3 international guidelines on biofilm-based wound care. Biofilms are present in over 80% of all of the chronic wound biopsies that we and others in our collaborative international group have looked at. So biofilm control is becoming a recognized essential component of chronic wound care. The reason these cause such a problem in healing skin wounds is because, as I showed you, they lead to chronic inflammation with elevated persistent levels of proteases and reactive oxygen species that cause tissue damage and prevent the wounds from healing. Solution and fibers containing the pterolidine are effective at killing the planktonic bacteria. We've looked at multiple species, and again, that's the concept of preventing the biofilms from ever forming in the first place. But also, once biofilms have formed using this pigskin explant model for skin chronic wounds, we can show that some of the formulations that Dr. DeLucchio has worked through are able to totally kill these mature biofilms. And as I said, that is a very important, unique distinction between almost all of the other wound treatment available. And I think that this taurolidine has a very important potential to become a very effective antimicrobial, killing planktonic bacteria or killing established biofilms, and critically important, it has such low cytotoxicity. Because to tell you the truth, we can take bleach from under your sink and pour it on biofilms and kill every biofilm. We've also killed every wound cell. So we have to have something that is effective against the planktonic and biofilms, but is not so darn cytotoxic that we end up killing every wound cell and impairing healing through that process. So I'll stop there.
Now you know why I'm so excited over a 7-year period. I've been involved with the company with a little hiatus, though, over the past couple of years, but since 2010, this really is groundbreaking. And to have something which is bleach, but it's both it can be metabolized, and at the same time, when it's acting in the catheter in between dialysis sessions, it kills all the bad stuff and leaves all the good stuff around. That's a pretty amazing thing. So I'm glad that you're confirming that from a basic science and cellular perspective. The next speaker is Prabir Roy Chowdhury, who is a professor of medicine at the University of Arizona Health Science Center. At any given time, he wears many hats and several suits, ties, shoes. And he's director of the Division of Nephrology, director of the Arizona Kidney and Vascular Center at the College of Medicine. He's a clinician with an active transplant practice, directs clinical and NIH-sponsored research initiatives with many basic science components. This has yielded over 150 peer-reviewed publications. He is co-primary investigator of our Neutralin Phase 3 trial. He's also internationally trained and has seen many healthcare systems up close. Dr. Chowdhury is actively involved in public policy and administrative aspects of dialysis, vascular access care, and hemodialysis as a board member, counselor, committee chair for a plethora of policy bodies. I have shared the podium with him at several previous conferences, and he always has something noteworthy to say. His unusually broad experience puts him in a position to see things from the top down and from the bottom up. He will share his perspective on dialysis healthcare with us and the need for further innovation in this space, especially in catheter care. Thank you.
Great. Well, thank you very much for that kind introduction. I really would like to thank CorMedix, particularly Tony and Koso, for inviting me to give this presentation. Do I just go to the next? Perfect. All right. All right, so what I'm going to do over the next 10 or 12 minutes is basically to divide my presentation into 3 main parts. So I will initially really make for all of you the case that we're not doing very well in vascular access care, and also make the case that one way to address this problem is to really go towards a model where we not only have an individualized vascular access care paradigm, but where we also focus on patient-centric innovation. In the second part of my presentation, I'm really going to demonstrate to you that I do believe that there is in fact a convergence currently of both clinical and financial imperatives that could actually incentivize these changes, reference individualization of care and patient-centric innovation. And then in the last part of my presentation, I'm really going to speak a little bit about central venous catheter infections, particularly on what I think could be a really important role for CorMedix's vascular access portfolio. And I was just very, very impressed by some of the data that Dr. Schultz showed about the impact on biofilm. I want to start off really by talking a little bit about value, and Koso in his initial remarks mentioned value as well. And I would argue that we currently as a community provide very poor value for patients with dialysis vascular access. And I say that because if you think about arteriovenous access, fistulae and grafts, our outcomes are, I think, extremely dismal. So we get excited about a primary patency rate that crosses 50% at 1 year. And we have, as you've heard already, a very high incidence of central venous catheter infections. And the problem, of course, is that in order to achieve these pretty terrible outcomes, we actually spend a very large amount of money, currently over $1.5 billion per year in this country, and this is without any mention at all or any focus at all on things like quality of life. So I think that the question that we need to answer as a community is, well, how can we break this cycle of a relative lack of innovation? And listening to the other talks, I think that that is being broken today. But the cycle continues in that it's a lack of innovation, poor quality and outcomes, and a very high cost burden. And I'm going to argue that two ways that perhaps we could break this cycle is by focusing on individualization of care, so really a precision medicine type approach, and number two, on patient-centric innovation, or by focusing on patient-centric innovation. And I'll talk a little bit about both of these. So what about individualization of care? I think that in the vascular access world, we absolutely have to get away from a one-size-fits-all paradigm that I think we currently function under, which is, let's get a fistula into everybody, and then once we've got it into everybody, let's try and decide whether or not it actually works. What I would like to see in the future is a setting or a care paradigm where we're actually stratifying our patients based, in my mind, not just on clinical, but also on biological parameters, and then individualizing vascular access care by pairing specific future novel therapies with specific clinical or biological phenotypes so that we can get the optimal results that really I think our patients deserve. So let's just give you two examples. In the future, if you have a 75-year-old diabetic with poor veins and two previous failed arteriovenous fistulae, I would argue that instead of putting in a third fistulae or even a graft, one should think about placing a long-term central venous catheter. The problem in doing this, of course, is that these catheters do terribly in terms of infection and thrombosis and poor dialysis and central venous stenosis. But let's suppose that in the future we had a new catheter which was a CVC plus, and the plus perhaps was a catheter that had a novel anti-infective therapy. Well, then the whole risk-benefit ratio of placing a catheter in this patient changes, and that could very well be a very optimal type of therapy for this patient. A second example could be an 85-year-old who has a poor life expectancy. Well, maybe this is not a patient who needs to get either a fistula or a graft, and perhaps the answer for this patient would again be another of these long-term CVC pluses, and the plus in this case potentially could be a coating, and I made this up, that prevents infection and thrombosis and central vein stenosis. And of course, I think it would be very important to identify who the best patients are for receiving these types of CVC plus catheters. The second thing I want to talk about, and I really think that this is important, is about patient-centric innovation. So what is this? Well, this is basically innovation that tries to target the issues that are important to the patient, not necessarily the issues that are felt to be important by the physician or the payer or the regulator or by industry. And the reason that this is so important is that there is a lot of data, particularly in the dialysis and even in the dialysis vascular access world that's coming out, and clearly in other areas as well, that a patient's perspective on what is important to them is often very different from that of the physician, the payer, the regulator, or the industry partner. So just to give you some examples, as a physician who's interested in vascular access, I've always said that it's about patency, and even more patency, and then infection, infection, infection. But if you're a patient, I would argue very strongly that what patients are really interested in are actually very different. So if you're a patient, you don't want the needles that go in 312 times a year actually to be painful. So cannulation is very important. And for us as a group of innovators and physicians, it probably means that we need to develop better needles. If you're a patient, not having to get admitted into hospital repeatedly is probably very, very important. So again, for us as a group, that would mean, well, maybe we need to develop better catheters that don't get infected and don't put patients into the ICU. And again, if you're a patient, you really don't want to keep going to the outpatient vascular access center for getting yet another angioplasty. And again, for a group such as ours, I think what that means is we need to develop a way of doing angioplasties and vascular access that doesn't result in this aggressive restenosis. When I put this slide together and I thought about these three indications, I also thought that there is one way in which you could use a disruptive technology, perhaps, to target all three of these patient-centric factors, and that is that if in fact we were able to develop a catheter that never got infected, never got thrombosed, never caused central stenosis, and I think very importantly allowed patients to take a shower, then suddenly this could become the optimal mode of dialysis vascular access. And again, I would argue that Neutrolin is, I think, taking some initial steps in this direction. So at this point, I'm going to switch gears a little bit and talk a little and really share with you some of my thoughts about how changes in healthcare could actually incentivize the things that I've spoken about, the patient-centric innovation and the individualization of care. So I want to start off by saying, and I think this is self-evident to everybody in this room, that healthcare is changing. We're moving from a volume-based system to a value-based system. We're moving from taking care of the individual to taking care of populations. And we're clearly moving from a fee-for-service system to a much more global payment system. I want to talk a little bit more about global payment systems because I think that's particularly relevant to the care of patients with end-stage renal disease. There are currently 37 global payment systems nationally, and they're called the ESCOs, or the ESRD Seamless Care Organizations. And these are basically a combination usually of a large dialysis organization, DaVita, Fresenius, DCI, a physician practice, usually a large community group, and a large hospital group. And an example, I think, is in New York is the one associated with Cornell. And what happens over here is that this combination is given a fixed sum of money to look after overall health care for usually between 500 to 1,000 hemodialysis patients. And when I say overall care, it is overall care. So if somebody gets admitted into the ICU and they're part of this ESCO, the ESCO has to pay for the cost of their ICU admissions, even if it's for heart failure. If you are able to take care of these patients for less than the amount of money that is given to you by CMS, you share in the profit. If you end up taking care of these patients for more money than is given to you, then you actually take a hit. And of course, you have to achieve quality benchmarks that are not only in terms of survival and adequacy, but I think very importantly for this audience in the area of vascular access. I actually looked up the different benchmarks, and I would say that between 20 to 30% of the benchmarks for the initial ESCOs at least were in the area of vascular access. The reason all of this is important is that I think one could make a very strong case that global payment systems could actually incentivize innovation. So in the traditional fee-for-service system, if, for example, you have a product that enhances fistula maturation or decreases central venous catheter infections, it may not actually be financially viable. And I've gone through this with other companies, and that's because in a very paradoxical and somewhat cynical way, in our current health system, caring for the complications of inadequate therapies actually generates revenue. And every time you get a catheter-related bloodstream infection, you're probably paying upwards of $20,000 in terms of healthcare. But in the ESCO world, or in the global payments world, this $20,000 to $30,000 that's spent on infection, for example, would reduce profit. And in particular, I would argue that if you could identify a priori which patients would benefit the most from your novel therapy, you could be in a very strong position in that developing, let's say, a $3,000 device that enhances fistula maturation, particularly in high-risk patients. If this could allow you to save a significant amount of money downstream, then suddenly the product is extremely attractive. Finally, in the last part of my presentation, I'm going to speak a little bit about infections and central venous catheters. And I really want to emphasize the slide on the left, which makes the point that as of 2 or 3 years ago, infections are now the leading cause of hospitalization in hemodialysis patients, having just overtaken admissions for cardiovascular disease. And that's actually, as a nephrologist, saying quite a lot. And I think it was because of this data that about 1.5 years ago, the American Society of Nephrology partnered with the CDC to start off a new initiative headed by Alan Kleiger from Yale called Nephrologists Transforming Dialysis Safety. And I would argue that just the presence of such an initiative clearly makes the point about how important infection and taurolidine and all the things that we've been talking about today actually are to nephrology as a whole. This slide just makes the point that central venous catheters are really not going to go away. So we're all familiar with the fact that if you start 100 patients onto hemodialysis, that 80 of them actually start with a tunnel dialysis catheter. What we perhaps may not be as aware of is that if you look out at 6 months, about half these patients still have tunnel dialysis catheters, and obviously out at a year, you're pretty much down to your normal prevalence of about 20%. The point that is important from this slide, though, is that we still need to take care of all of these catheters. And so a product like Neutrolin or taurolidine clearly could be very important. If you look at the slide on the right, it just basically makes the point that the prevalence rate of central venous catheters, whether they're more than 90 days in place or less than 90 days, has pretty much remained stable. So clearly there is an unmet clinical need because, as Gary mentioned right in the beginning, if you have a catheter, you're likely to get an infection. Tony's going to speak later on about Candida auris, and I've put this slide in really just to make the point that one always has to think about emerging infections. One always has to think about resistance. And again, from the time that I've known about taurolidine, and I did hear about the initial data that Gary spoke about huge amounts being infused, but I was always fascinated by the fact that it's not an antibiotic and it's across a wide range of different microbes. So I think that that's actually a very important point. So how does one put all of this together? So I would argue that if you think about all the things that I've said, the unmet clinical need, the financial imperative, the need for a more precision medicine type of individualization of care approach, and this focus on patient-centric innovation. If you put it all together, I would argue that a product like Neutrolin that reduces bloodstream infections across a whole range of different microbes, and also very, very importantly, seems to be effective in reducing biofilm formation, can actually play a very important role in targeting these different things that I've spoken about. I want to end by making two personal points. So, and again, these are both personal messages. So, number one, when you're thinking about innovation, particularly vascular access innovation, it's really best done through a team approach. And your team needs to include not just clinicians and industry partners, but I think also very, very importantly for the dialysis vascular access world, large dialysis organizations, and also hospitals and healthcare provider organizations. And then number four, and very, very importantly, the partnership needs to involve federal agencies. I've learned so much in the last 5 years through my association with the Kidney Health Initiative, which is a public-private partnership between the American Society of Nephrology and the FDA. And I would argue that when you're talking about federal agencies, it's not just the FDA that we've spoken a lot about, I think very importantly, it's CMS, but it's also NIH and it's also CDC. If you really want to do innovation, whether it's product innovation or process of care innovation, I think you also have to involve your patient. I think you need to know what the patient factors are. You need to know what are the things that are important for patients, and you need to get feedback from patients once you've actually developed your therapy. My second message is that if in fact as a community we want to be serious about breaking this vicious circle of a lack of innovation, poor quality, and a high cost burden, then I think that we need to try and develop a new construct. And I would argue that that construct should be a construct where we try and use patient-centric innovation and a more precision medicine, individualization of care approach to really target both the unmet clinical need and I think the financial imperative that is currently there both for dialysis and for dialysis vascular access. And again, I would argue, based a lot actually on what I've heard this morning, that a product like Neutrolin could actually have a sweet spot in this future healthcare paradigm. So again, I'm very glad to be involved. I appreciate the invitation. I'd be happy to take any questions. Thank you. Tony, actually.
Thank you, Prabir, and one statement that always resounds in my mind is that we're here today because we're standing on the shoulders of giants, and we're honored to have Prabir as our co-PI with Michael on, and he's been very helpful to advancing the study. I also want to recognize we have several of our PIs here who are from very high enrolling sites who support the study, and our major mission and focus is the study. These are foundational sciences for the study. And to get to the last topic before Q&A, I've been reminded to stay on time by Josh. The development of novel infections. So we know about bacteria, MRSA, VRF, Pseudomonas that's resistant. We also know that taurolidine works against clinically relevant fungi, which are fungi that spread quickly throughout the hospital. And one particular one, which we were made aware of by CDC recently, which has been a growing concern, is Candida auris. Now, Candida auris is essentially a very virulent and resistant strain of Candida, and it's spread through contact, and it involves patients who have had previous treatments for infection as well as hospitalizations. Frequent, and also patients with central venous catheters are a high-risk group for Candida auris. So we studied this recently, and we have several publications in the works on it. We worked in conjunction with JMI Labs, Jones Molecular Institute. It's a microbiology lab that is a fabled lab that's well known throughout industry and by regulatory authorities. There are reference labs. So JMI Labs essentially did some additional studies. This yeast is highly resistant. So it's resistant to amphotericin, diflucan or fluconazole, and voriconazole. And those are the three most prevalent antifungals used. It was first identified in 2009 in Japan. I think it was first isolated in South Korea about a decade before, actually 1996. And this is an important, 1 in 3 patients with Candida auris die, so it's very dangerous. So we went about seeing some of the risk factors, ICU, which is one of the indications we'll eventually be looking for, central venous catheters, which involve dialysis, as well as ICU, and oncology, history of infections, as I mentioned, and the resistance. And also it forms biofilms, which enhances its pathogenicity. These are the references here. Okay, if you can see the map of the United States here, it's most prevalent in these areas. So right in New York here, we have a lot of incidents, and it's in New Jersey and also in Indiana and Illinois. Okay. That's a good question. I think that there was an original cluster in New York which spread and it wasn't contained quickly. Basically, we did studies that show 100% of taurolidine in vitro, essentially 100% of Candida auris strains, which were CDC strains. So these were very well-characterized strains of Candida auris from the Centers for Disease Control, which were sent to Iowa, where Jones Molecular Institute is, were sensitive to taurolidine at the concentration found in Neutralin. So obviously we wanted to make sure it wasn't a super concentration of taurolidine. It was consistent with the concentration, the 3.5% found in Neutralin. Essentially, we believe that this could be helpful in the future. We've informed both CDC and FDA about it and sent them communications about the efficacy of taurolidine in addressing Candida auris. So I think I lived up to my commitment to remain on time, and now we're at Q&A, and both Gary, I think, and Koso will address the audience.
Thank you. Questions? Yes, go ahead.
These are for Dr. Pfaffel. With respect to the strains that were assessed in the taurolidine studies with Candida auris, do you have direct indications as to how many existing marketed drugs these strains were resistant to? Did that cover strains that were known to be resistant to fluconazole and voriconazole and things like that? And secondly, how quickly, and I think you and I have discussed this in the past briefly, how quickly do you think Candida auris could truly manifest itself as a significant public health hazard in the U.S.? I mean, because obviously it's so far, at least thankfully, apparently been localized to a specific region. So maybe you could give us a sense of what public health physicians are looking at and what they are thinking about how long before it becomes a real public health problem in the U.S. Thanks.
Okay, I'm going to address that briefly and then hand the microphone to Bruce Reidenberg, who's did a lot of the work on these publications that we're working on and the data. So to answer your question, there was a fulsome analysis of taurolidine versus Candida auris on many isolates, and those essential Candida auris has been tested against other antifungal agents, and it ran the whole gamut from the conazoles to also the amphotericins, as well as the other, the Merck agent, which is a direct cell wall penetrator, and I'll remember the name of it in a second. And it is very easy to transmit, and it's mostly by hand contact. So using hygienic standards and aseptic technique is obviously key, but it's really easy for nurses in the ICU and other critical care units. And dialysis unit is considered a critical care unit of sorts to transmit this. So it is so far, because the CDC seems to be very aggressive at sending the word out, it's been contained, but the number of cases keep growing. So Bruce, you want to comment?
Thank you. So the particular strains that we tested that were received from the CDC were resistant to amphotericin, fluconazole, voriconazole, and capsofungin. As far as the public health issue goes, hand washing is of course critical, and for hundreds of years we've known that to be the case, and it's still difficult to implement. And most of these patients are quite debilitated in the ICU, and as an ICU nurse has 2 or 3 patients as her responsibility, and the ventilator starts alarming, if they're working on a person who has Candida and the ventilator's alarming, they don't have time to wash their hands. They're going to touch the ventilator. And the issue of not cleaning that ventilator surface is really what brings it to the next person. Candida is particularly strong at managing dry surfaces. And so I think it is a significant threat. However, if you look at how methicillin-resistant Staph aureus took so long to penetrate through the United States, I think it's unlikely it's going to happen quickly. But but I think without strong vigilance, it is going to happen. Now, one of the things that Dr. Chaudhary mentioned that was very important is the new economic incentives. And so the hand washing is going to fit with new policy changes to increase the number of staff. It's probably going to be scolding staff to say, wash your hands, wash your hands, wash your hands, as opposed to an extra nurse so that the ventilator gets controlled more quickly. But I think that pressure is going to decrease the public health threat that we perceive right now. Nevertheless, for Neutralin, there's no difference. Whichever bacteria evolves whatever mechanism of resistance, the Neutralin breakdown into these chemically active moieties is going to continue to destroy the external surfaces of the bacteria or the yeast, and even potentially free virus. So there's a lot of potential still, even though we hope to have better public health in the near future.
To some extent, it's apples and oranges. We're comparing a disinfectant and something that will inevitably or absolutely prevent a catheter lumen from being infected against other antibiotics, which then treat them within the body once they've established infection. So, what we're demonstrating is not that we're treating this or that we're going to avoid any type of epidemic in the future that may happen for whatever reason. We're saying it's not going to be the catheter. We won't let this candida get through the catheter to get into the patient. And I think that's a fundamental accomplishment when we all know that catheters are the source of many of these type of infections as a vehicle to get into the body. Okay, any other questions? Well, I'd like to thank all the presenters. It was really enjoyable and fine and on time. And I'd like to call back Koso for some finishing remarks. Thank you.
Well, thank you very much, the moderator, Gary, and all the speakers. I think we took a great opportunity not just to look at basic science, but also to bring it to life, to see in the variety of different settings, the variety of different tools that we would be able to take our platform and leverage it. So that is going to be critically important. Some of the key points that I'd like to make, if you look, and what excites me a lot, and I think was mentioned a couple of times, is when you look at our pipeline, if you look at the medical devices, what was covered on sutures, on hydrogels, on meshes, you can see it takes very little amount of resources to actually develop this, and it can move very, very fast. And that's critically important. The opportunity is enormous. You saw the worldwide market of about $8 billion US dollars. The US market itself about $4 billion US dollars. So it's critically important, and the resources required is very little. The beauty in our focus, and we talked about partnerships, is to ensure that we network. We've networked with a lot of universities, a lot of individuals who have had a lot of love for CorMedix, enjoy working with CorMedix, enjoy taurolidine and where we can take it. And that has been critical in our ability to move forward with the speed we have. If you look at neuroblastoma, which we've talked about, and our efforts with POET, here in the United States, we've got 70,000 kids per year that suffer with neuroblastoma. It is a devastating disease, and if we are able, with taurolidine, to help make their life better and improve the chances, it would be a major step forward. So that excites me a lot. When you look at Neutralin, which someone described here as our first love, which is very true, some of the efforts with biofilm, mature biofilm, C. aureus, again begins to build the properties of taurolidine and shows once we've completed our phase 3 studies, we get the FDA approval and we can bring it to the market, you can see the properties that Neutralin will have and what it can achieve. So that is what excites me a lot. So here's our pipeline. We have Neutralin in Europe. Through our partnerships, we will continue to grow Neutralin in Europe and the Middle East. We are continuing to drive our phase 3 studies here in the US, and we will provide update to that as we have future updates. In the meantime, for a fraction of the cost of what it cost us to do the Neutralin studies, we are moving our pipeline forward, which will help CorMedix overall as a company and will help our shareholders. Thank you very much for all the insightful questions you had and for participating in this meeting. We look forward to providing you updates as we go forward. Thank you, and safe journey back for those of you who are here. Thank you.